Yes? Okay, we can start. Okay, so good afternoon. It's time, and we start, and it's really with great satisfaction that we welcome you here for this symposium, Dangerous Liaison on Emerging Viruses at the Human-Animal Environmental Interface. This symposium is organized in the context of the anniversary of the celebration of our birthday as HKU-Pasteur Research Pole, a joint laboratory of the Institut Pasteur and the University of Hong Kong, of the Li Ka-shing Faculty of Medicine of the University of Hong Kong, which was established 20 years ago. And so we had thought of having perhaps a different type of symposium on this occasion, but because of the travel restriction due to the coronavirus, we have opted to spread a series of events throughout this year, and this is the first scientific one after our anniversary party at the end of October. So why are we talking today about emerging viruses and at the human-animal environmental interface? Well, not just because there has been a pandemic for over a year due to a new coronavirus, but also because this is really what we know how to do here in Hong Kong and at HKU-Pasteur. Because of this synergistic approach, together with the group at Hong Kong U, which has pioneered research on respiratory viruses for, I think, over 25 years, we have now been able to collaborate and produce results at this interface, working not only from the SARS outbreak in 2003, but also on the H1 pandemic in 2009, and then started working again on coronaviruses after MERS, and then the avian influenza outbreaks in China, H7 and 9, and now, of course, in 2020, the coronavirus. So this infectious disease at animal-human interface in particular is one of the major strengths of our laboratory, and we thought that because of what is going on now in the world, it was really a topical issue to be discussed at this workshop. And more recently, we have started also to add to this groups working on antimicrobial resistance, microbiome, microbiology, and genomic. So HKU-Pasteur at 20 has now 5 laboratories working together on these topics, about 45 staff, many publications, many collaborations and links, and this is what we need really to thank also for the organization of this workshop. But why did we choose the Dangerous Liaison title? Well, first of all, it's a novel by a French author, so HKU-Pasteur, perhaps it was fitting to include something like that. But because also this novel tells the story of 2 very manipulative individuals who are trying to exploit and control the life of other people. In fact, they are poisonous. And this is exactly what has happened to us for the past year when, you know, a small, not even organism, but a small particle of life composed of RNA and surrounded by membrane has clearly appended many of the values and the way of life that we have known thus far. A virus is also the Latin word for poison. So this is why we thought it would be really a good title to express the fact that we are always in a delicate balance between these different components of life on our planet, the environment, the animals, and us as different kind of animals, but still animals. So it is important, therefore, that we try to explore what have been the consequences of this pandemic, not just for health, not just for public health, but also for society, for research. And therefore, to have over these 2 days an integrated view with inputs from different research fields to try to project what will be the next level of research that we need to implement in order to be better prepared, in order to continue our approach to mitigate the consequences of these zoonotic events. I think that we all agree that the language of risk, of which we have now really become quite acquainted, changes the way that scientific research is conducted. And also, society and the media raise questions that cannot be addressed only in the lab. And this is why it is important to discuss not just about scientific results in terms of papers published in our prestigious or less prestigious journals. Think about it. 100,000 papers have been published. If you Google or if you go to PubMed or Medline with COVID-19 and SARS-CoV-2. So perhaps it's time also we start thinking about something else and move forward and try to decipher what we need to do. Because this is not the first time that we had a pandemic. We had pandemics, I'm not talking about medieval times, bubonic plague, etc., and not even about the so-called Spanish flu. I'm just talking about 1957, the Asian flu. I'm talking about 1968, the so-called Hong Kong flu. There too, millions of people died, although probably we don't have the right numbers. So what was different then? Were the healthcare systems better? Was society reacting differently to that? Was the timeline of the spread of the virus, because we were not traveling that fast and that much, that changed our perception? These are issues that, as society, we need to address when we're doing research. And therefore, I think we're very fortunate to have these 2 days of discussions, which will be started with a keynote lecture, of which in a moment, and then we'll have also panel discussions with points of view from specialists in these fields, and then hopefully a very open, candid discussion about what are the important matters at stake at the moment. And I would like to conclude by thanking in particular, of course, the sponsors, the Pasteur International Network, the Pasteur Foundation Asia, that have helped us organizing this symposium, and the people involved, in particular Vijay Dhanasekaran, Mathilde Boisserin, Simon, Caroline, Jimmy, and I think many other who have contributed to making this event possible. So I think that I'm on time and ready to thank you again for being present here today, and to move to the introduction of our first keynote speaker, who is Professor Marion Koopmans. Marion Koopmans is the director of the Department of Viral Science at the Erasmus Medical Center in Rotterdam, and I'm sure all of you know that this is one of the largest and most important clusters of excellence of research in viruses, many types of viruses. She graduated as a veterinarian in the Netherlands, spent also time training in the United States, in particular also at the CDC, US CDC in Atlanta, and then came back to Europe, I think in the Netherlands also, to work at the National Public Health Laboratory, where she conducted important research, also trying to, let's say, extend the laboratory results to public health policy and practice. And in the Netherlands, she has been acquainted with outbreaks at the animal and human interface since the H7N7 outbreak of avian flu, I think about 20 years ago, if I remember well. And then since 2006, she has been appointed as full professor at the Erasmus Medical Center. Among many other things, she's also leading one large consortium of the Horizon 2020 in Europe called COMPARE. And of course, she's one of the SAGE strategic advisory group experts for the WHO. And in this capacity, she went, in fact, to Wuhan as one of the components members of the important mission on the origin of coronavirus. So it is really fitting that the first day on coronaviruses would start with her talk. And I think that with this, I would thank you for your attention, remind you that after her talk, we will have questions from the audience, and then many other people, colleagues are joining online, and we will try to give them the time to talk and ask questions directly, or if you want to write your questions on the chat, we will read them and ask Marion to answer later. So with this, I stop sharing my screen. Here we go. And there is Marion. Thank you very much. The floor is yours. Thank you for accepting our invitation.
Good morning, and thank you very much. Well, let me start by saying happy birthday for the Institute collaboration and happy New Year. And thank you very much for this kind invitation. I personally feel the world has, if I must say one positive thing about this pandemic, the world has become smaller, because I think I'm on phone calls with Malik and colleagues on a very regular basis, for instance, through the WHO work. So it's my great pleasure to present here. At first thought, I was like, what am I going to present there? Because these guys know everything. But I decided to focus on an element in this pandemic that I have found very intriguing, and that I hope will help us also in our endeavor to try and explore where this virus comes from. So what happened at the origin of this pandemic? And that's insight from the genomics field, full-fledged now part of the whole pandemic research and response. And yes, indeed, this is focusing on the human-animal interface. This is a sort of a visual that we made for that Horizon 2020 consortium project, just to keep us constantly reminding of how important it is to not just have an anthropocentric view, but to consider us as part of a wider ecosystem which we share with animals and with pathogens, and which is under pressure because of everything we do. Sort of the nature strikes back. So what I will do is also focus on what we've learned from the work at the human-animal interface in this. But starting at the start, so very much in line with what has been happening over the past few years in outbreaks of, for instance, Ebola. In this pandemic, we've seen the very fast forwarding of genomic sequencing as part of the outbreak response. And this is no exception in our country. So what we did, like many others, is develop a workflow that would allow real-time sequencing with 24 to 48-hour turnaround time, so that it actually could inform the public health decision-making. This is, of course, not something that you can do. It goes beyond the sequencing. It does require strong partnership, as you have at in your institute, with people in the hospitals, with the public health experts, to get reliable metadata with sequence data and get meaningful outcomes for that. Now, what that required in our hands is to develop bioinformatic workflows that allow that speed, because it's one thing to be able to do a rapid turnaround sequence for one sample, but this becomes a very different ballgame if you want to do that on a routine basis every day. So this was work done by postdocs and PhD students on the fly during the initial stages of the pandemic. And what I think is also quite fascinating and new in this particular pandemic is just the wide sharing of data through GISAID and other platforms, and how that has helped inform our public health response. So what I will talk you through is a bit of how we used this in the initial phase of the pandemic, and then also in relation to understanding what is going on at the human-animal interface. So this was at two levels. Of course, pandemic became visible through disease notifications, people hospitalized, and that raised a lot of questions around hospital hygiene, for instance, clustering and hospital hygiene, and also at the community level. This is a first experience that we had. So for us, the pandemic started almost 2 years ago, 1 year ago, for the Netherlands, I mean, with the first case identified, recognized, I must say, February 26th. So that is tomorrow, I think, yeah. And that was a traveler from a ski vacation who had come back. But in the days after that, when this was discussed in a national outbreak response team meeting, there was already a representative from a hospital in the southern part of the country who said, I don't know, I have a hunch there's so many people in my region that seem to have colds. I'm not sure what's going on. So what we did in the course of one weekend is screen healthcare workers with very mild symptoms from 10 hospitals, more than 1,000 of them. And as it turned out, already 50 of them were actively infected with SARS-CoV-2. And the sequencing showed that these viruses were already all over the place, with here the central virus, including the reference virus, the Wuhan 1 strain. So already widespread circulation, completely under the radar, before we at the time when we identified the first severe case. And this immediately led to the decision to go into lockdown for a big part of the country. Now, this was related to two events. One was the winter sports vacation, which was really around that time, which is an extremely popular holiday in our region and in Europe. So there were millions of people actually enjoying winter sports in Austria, Switzerland, and Italy. And in Italy at that time, of course, there was the northern Italy Bergamo situation where this pandemic was really exploding around that time. And the second part was that the part of the country that did not go on winter sports was doing Carnival, which also is, as you can see, a very popular holiday with people visiting bars for parties. And the tradition there is also to go from one place to the next with groups. So perfect spreading event. Now, that immediately triggered lots of questions. So we saw a lot of healthcare workers infected. Was that particular to this group? Was something going on in the hospitals? So immediately after that, there was a flurry of questions around clustering of, so do we see clusters of SARS-CoV-2 in healthcare? Is this amplification because of the situation in healthcare? And you see here some examples of a mixture of patients, so the squares and the triangles of patients, and squares of healthcare workers. Clearly a cluster here in this particular hospital, and here, but these were actually the exceptions that we saw, because once the realization was there and proper PPE was installed, we rarely saw clusters of infection in the hospital setting anymore, and that was very important because of the concerns around this. What's also quite clear and now documented widely is that with genomics, you can actually really learn additional things. And here's an example of an outbreak that was, all over the news because it was seen as evidence that church meetings and singing was a high-risk activity. And this was an outbreak in a nursing home with an on-site chapel, where visitors of a church service in that chapel became ill, and then there was a large subsequent outbreak in residents and staff of that particular long-term care facility, which then was closed for visitors, and so was the chapel. And there was a lot of accusations in the media about that. But then adding in the sequencing, what turned out is that this was actually a very mixed pattern. So there were, these are all sort of zoom-ins from a large, by then already large phylogenetic tree, is that there were quite a few sporadic cases in there. There was small clusters, and there was a mixture of sporadic cases interspersed with community cases from that same region. So what this actually told us is that this was more like the community-wide emergence and spread of the pandemic that became visible through people visiting relatives in the long-term care facility, with maybe a little bit of amplification, but that was really not the full thing. That included the people that that had infection that visited the church, the chapel, because they were all over the place here. So more a reflection of widespread circulation in the community that you see once you zoom in and do an outbreak investigation than that it was a real outbreak. Similar applications in, for instance, the meat processing industry. There has been a lot of talk about that in meat processing plants, which had clusters. There's examples here from Germany. There were reports from the US. We've had reports from the Netherlands. And there, again, in the media, this was linked to the working conditions in these plants, and particularly the fact that many of the people employed there were migrant workers with their own specific housing facilities that may or may not be favorable for virus transmission. So here's what we saw there is, this is an investigation again in one of the big meat processing plants, where we saw in red here a large cluster of sequences from employees. Sorry, this says employers, it should read employees, who were actually from very different parts of the region. They lived separately. They had separate transportation to the facility. All that they shared was the facility itself. So clearly not explained here by the housing conditions of these individuals, but by the work in the facility itself. It also showed that there were separate introductions in that facility. Here again, with this introduction, again, separate employees, separate housing, separate transportation, but there was also small clusters that were from the same household. So a mixed pattern, and really showing that you need to be careful in jumping to conclusions here, which is what happened a lot in our media anyway. Then moving to the human-animal interface, the area of interest that I really hope now will that focus will stay with us and will get the attention that it deserves. I remember that Malik and I were involved in trying to put forward a proposition to the World Health Organization to say, okay, please develop centers of excellence with expertise on the human-animal interface. Let us propose a network around this with funding so that we can collaborate and prepare for these kinds of events in the years to come. That was a proposal as part of the R&D Blueprint initiative. So far, I don't think we have gotten the ear, but I do think we have to retry that again in the coming period. But this is looking now at the human-animal interface, and the first evidence of infections in animals, of course, coming from Hong Kong, how apt. With dogs infected, also first indication of cats infected from experimental studies, from clinical observations, and from the antibody surveys in Wuhan. The observation of infection in tigers and lions in the New York Zoo, where you still wonder how on earth did they get infected, because that's not an animal that you approach very closely, I would say. And then based on that, we had a national outbreak management team meeting to focus on the question about potential for animals involvement. With the pets, of course, with the question, is that a risk for further transmission amplification among people? But then also asking ourselves, is there other animal species that we should be worried about? And that brought about the attention to mink because of their relatedness, taxonomic relatedness with ferrets, that of course we were using as animal models. And that's what brought about a screening program for mink farms. So end of April, we had the first positive farm for positive animals testing positive. And this was part of a program where farmers sent animals that die to an animal health service for pathology investigation. That's just a routine program. And because of the size of these farms, there's a couple of dead animals on a weekly basis. So that's just that was the starting point. So soon after that, we started to see quite a few additional positive farms. So that formed a One Health consortium to really investigate this. So what we've seen is just how fast and easy this virus spreads among mink. This now is the number of infected farms, as you can see, that spread out through the year because it simply was unstoppable in our hands. The immediate national response was to go for culling of infected farms and for movement restrictions between farms. But regardless of what happened, it was impossible to stop the spread. So eventually the decision was to get rid of the entire industry. Infected with different clusters of viruses. I will talk about that a little bit more in these colors here. Most of the detections were based on clinical signs or routine monitoring. An important message here is that not always, clinical signs were not always evident. So it's difficult to recognize. So some farms were picked up as infected through serology screening, and they were localized in this region of the country, which does have density of all sorts of farming. This is now the genomic information in combined with all the human sequence data that we had collected by that time. And as you can see, there were these separate clusters here, A, B, C, D, and E. And that's the reflection of separate introduction of the virus on these mink farms. And from then onward transmission between farms. And of course, an immediate question there was for also people working or living in the towns or villages around those farms, is there onward transmission in the community? So what we did is pull notification samples from people that were notified with SARS-CoV from those neighborhoods from the same postal code areas. Those are the purple bars here. But we did not find any hits with the sequences as they became recognizable through animal passage. So that's unlike the situation in Denmark. But this situation kept on going. So this is a phylogenetic tree with in red the human sequences, in blue the animal sequences, reflecting here all sort of a representation of the genomes from people with the different clusters here. And here in triangles, the community cases. And as you can see, two of these clusters were larger than the other ones. Here's one that stopped quite easily. And particularly cluster A kept spreading quite heavily. What we then try to do, this is work together with Sam Lickett and Lulu from the University of Edinburgh, is to see if there was any indication that the evolution rate of this virus would be different when spreading on the mink farms versus people. And that's not, her conclusion is that there's no evidence for that. So similar evolutionary rate. And that was a question that we needed to try and time when this virus was introduced on these different farms. And that time to most recent common ancestor analysis was actually here on this slide. So we timed the introductions all to March, latest April. But from there, then farm to farm spread continuing through here through October. Now, with that farm to farm spread, of course, what you can expect as virologists is that leads to evolution of these viruses. And that's what we saw. There was an alert at some point in time of that from the mink farm situation. And this is something we've also observed in our outbreaks. And particularly, of course, the larger, the more transmission there is, the more risk of accumulation and evolution you see. And there's clear evidence of that in our cluster. So here again, you see the clusters. But now, and these are all aligned sequences focusing on the receptor binding region, where you see mutations, amino acid mutations, in sometimes temporary. For those of you that are following that closely, this is a mutation that was seen in the Danish cluster 5 sequences that spread quite widely into the community, occurring on separate occasions, so co-evolving mutation, but also other mutations, and particularly this one here and this one here that we saw. This is looking at that again now in a timed phylogenetic tree, where you can see here, this is a particular mutation of course, is a position that now is under scrutiny because of the emergence of the new variants with increased transmissibility. A mutation that popped up, but this particular lineage of viruses died out, possibly through the containment efforts there. Here's another example, and here's a third example of these mutations popping up and dying out. But this one is the one that really continued to spread, which of course you cannot say is because of this mutation, because it could also be because of peculiarities in that region or in the people around those farms, but that surely was a virus that kept spreading, and again with a mutation in the receptor binding domain. Now, the different mutations that I mentioned all are in contact residues with the ACE2 receptor. And of course, that is relevant to understand. We've gotten a lot of questions. Does this mean that you see evolution of variants in animals? I do think it's possible, but I do think as likely an explanation is that this is adaptation to the animal ACE2 receptor. And of course, it's hard to tell whether or not that reduces the risk for circulation among humans. That's studies that we're trying to do nowadays. What was interesting is that for that particularly 486 mutations, there is only very few of human genomes that have that particular variant in their genome, but it is found in the bat and pangolin genome. So that's an indication, according to us, that this is indeed an adaptation mutation back to animals. There's others there, and this is work that is also being discussed in the WHO virus evolution group that Malik and Leo are taking part in, where of course we are now trying to understand what all of this diversity means and does to the viruses as they continue to circulate. Back to molecular epidemiology. So of course this genomic sequencing was also done to try and understand transmission. So what we did is combine our cluster findings and the branching in the phylogenetic trees with a lot of the information from the track and trace situation, looking at was there shared staff or owners, was there anything shared in the feed supply, what was the potential role of stray animals in contact with these farms, environmental contamination, and potential role of wildlife. Well, and that is, and I want to point out a few of those things. First, of course, humans living and working on those farms, it is clear that that situation is an increased infection risk for those individuals, because what we saw is that more than half of the people that we tested tested positive for SARS-CoV-2, which is much higher than the prevalence, certainly also the seroprevalence that we have in the general community, which is around 10 to 50% now, but which was much lower around then. But that was also the case for cats and dogs. So because of the layout of these farms and the access to the mink food, they do attract semi-wild cats. So as part of the effort, we set up a protocol to capture them. And what was found is that there was infection in the feral cats and also in some of the dogs in the household. And in some of these feral cats, the virus was also detected, and those were then the viruses from the mink. So similar sequences to the mink viruses. We never found the virus in household cats on these farms, interestingly. So it was related to these feral cats. Question then is, do these cats move? Cats, and particularly male cats, are known to stray quite widely, and because of this high density farming region, that was an option. So that was looked at because some of the animals have been chipped, but it turns out that because of the permanent presence of food, they actually become quite lazy. So they really hang out around the farm. So so that was considered less likely as a vehicle for transmission between farms. But we actually decided to keep feeding them after the culling of the mink to also avoid them starting to move around. Here's a bit of the layout of the farms. So large halls with a lot of access and with quite densely packed animal cages here. So one part of the investigation was doing environmental sampling, both dust in different locations. So there was dust collecting devices here, but also air samplers on the bodies of the people working here. And of course, that showed that there was widespread environmental contamination in settled dust. Here on sort of environmental swipes, bedding material, fecal samples, and specific swabs. And the swipes, 100% of them was positive with really, really, really high viral loads or RNA loads. Bedding, often positive, fecal material around a half, swabs around a third, and with differences between the farms. So here's a farm where that the rate of positive was much higher than in the others. But from the genomic data, we could tell that we were earlier after the incursion on the farm here. There was less diversity at the time of sampling. So this really reflects the acute phase of infection on this particular farm. This is a busy table, but this is then looking at presence in dust. So the different in air, the inhalable fraction and PM10 fraction, and all you can say is that there was a clear presence of RNA. This is all PCR material. We have not been able to culture virus out of these samples, but in our hands, I know that you have experience there, and you're probably familiar with some of the work going on in our institute with Sander Hert. In our hands, culturing virus in these air sampling studies is actually quite difficult. Now, there has been some warning. There's a paper from the EcoHealth Alliance consortium that has also said, beware that there may be the risk of re-establishment of SARS-CoV-2 in wild animal reservoirs. And that's, I think, a concern through this type of passage. And they did map our country or our region as one of the areas where that risk is present. So we also looked at, that's also been observed, of course, around avian influenza outbreaks, where if you then sample wildlife, wild birds, we have observed spill back into the wild bird population. So we looked at what types of animals that could potentially be susceptible are around and where are they. Here's the region where the mink farming is, and you can see the different distributions of the animals. And Thijs Kuiken and his group has done testing of wherever we could find evidence of these animals. But so far nothing has been positive there except for 2 escaped mink. And that's a situation that is present. I have learned, I didn't know this, but in many of the countries that have large mink farm production, that there also is feral mink populations from escaped mink. The Netherlands is an exception there because we are too densely populated, so there's no ongoing risk of wild mink or feral mink transmission, but it does again point at the sort of volatility of this type of situation. So this is also what was put in a risk assessment, just appeared a few days ago, joint tripartite risk assessment saying, listen, hey, look out, there is a serious risk of introduction and onward circulation of these viruses in for animal populations, and that they strongly urge for screening for that. And I think, and this is my near last slide, that this is a stark reminder of just how we are outsmarted constantly, I think, by viruses. This is now a mammalian tree showing the different examples of animals that are have been documented to be susceptible for SARS-CoV-2. I'm sure it's not going to be the final picture, and we really need to have an open mind also in our studies looking at the origins of this pandemic. So that's what I would like to share, what I wanted to share with you. I'm now very much a fan of having genomic sequencing embedded in these kinds of studies, although the ramping up now is creating some headaches on how on earth we keep doing all of this. I think the spillover into fur animals is a concern, not so much for reservoir establishment because I think they are too susceptible. It's more like a wildfire, but because of the further spillovers that can happen because of the widespread environmental contamination. And so I am a strong supporter for this call for increased biosecurity and monitoring of that. And with that, let me thank the consortium that really did a lot of these studies, public health partners, veterinary partners, and of course, funders that allow us to do these types of things. And I'm happy to take questions.
Thank you very much, Marion, for the talk. And I would like to start with questions from the audience here in the lecture theater, if there are any. And let me also take a look at the raised hands. Let me see who would like to start. There is, of course, a microphone. Yes.
Thank you. Can the speaker hear okay?
Yes.
Oh, wonderful. You had a couple slides towards the end of your talk regarding some feral cats in reference to the mink population, and compared those to some house cats. And I wondered if you had any reference points about how the feral cats maybe have been contaminated compared to house cats. If you believe this is also due to the widespread environmental contamination or perhaps some other factor.
So you have to repeat the question. The moderator needs to repeat the question because it's very soft. I heard here it's about house cats versus feral cats, yes, and maybe environmental contamination.
Yes,
but I'm
correct.
Not sure if that...
Yeah, I just wanted to see if you knew the source of the contamination of the feral cats compared to house cats. I think it's quite an interesting occurrence.
So what was observed is that these feral cats, they actually, hang out, the farmers try to keep them out, but that's not very successful. So there was even, during inspections, there was nests of cats in the barns. So they really have close interactions there, because the way the animals are fed is through, a meat slurry that is put on top of the cages, which is there all day. So it's very easy access food. So that's what happens. And on those farms, there's this the dust is very heavily contaminated. So once in the barns, there's just ample opportunity for contact. The house cats are very actively kept out of the barns. It's amazing that is so separated off. Of course, the numbers are not huge, but so it is likely related to that type of exposure. It is interesting because people on the farms also were infected. So you would expect some transmission to house cats through people. We have seen that, but not in this situation. So we think it's related to just the amount of exposure.
But do you think that, if I may follow up on that it is due to the concentration also of animals in those barns? Or because, as you say, if the staff is contaminated, then they will also go home and have their own pets. So
That's true.
it is being amplified just by one random contamination.
That's true, but if so, we've done environmental sampling also in other environments, for instance, in the slaughterhouses, in nursing homes, in hospital, where it's difficult to find evidence for virus on environmental surfaces. That was not so. If you compare that with the situation on the farms, so you have dust with CT values of 20, massive contamination. Um, um, Also in the air just at the doors of the farm. So the level of contamination is so much higher in that kind of environment that I think that that is likely to be part of the explanation.
Just follow up briefly, just with one further comment there. So in terms of this widespread environmental contamination, I believe this is, as you just touched on, is largely related to the farms itself, correct? And I wondered if you had any
Yes.
knowledge of how this compares to other previous outbreaks, as our moderator, our speaker mentioned early on when we first began. For example, even just SARS in 2002 or other pandemics in the past. Have we seen also large contamination of environmental samples?
Have we seen that?
Yeah.
In
The environment.
so we have some evidence for avian flu outbreaks, where so we've had H7N7, large situation with H7N7 outbreaks in 2003, where we saw spillback into wild birds around the farms. And there was indeed very heavy environmental contamination, which was also then, it raised the questions, what do you do with the material coming out of the farm? So the decision then was made to not decontaminate, clean the farms right away, because that actually has quite a risk of dispersal, but to do that after a mandatory wait period for, I think it was like 40 days, and that was based on an assessment of the time needed to reduce infectivity.
Yes.
And something similar happened here. So the farms were left alone before the decontamination was done, which is very difficult to do without dispersal.
Has another question from KG here in the room.
Hi, Marianne. This is KG. Very nice talk. Thank you. Marianne, your slide from Equal Health Alliance suggests that the human pandemic is acting as a vector, as a mechanism for spreading the SARS-2 virus to other animal species and reestablishing it. Is the primary concern that you are creating reservoirs for the virus to continue to infect people, or is the primary concern that you're driving the genetic diversity and creating more dangerous strains?
So our primary concern with that led to the decision to recommend culling was the second. So we were dealing with response to pandemic. We had more infections in animals than in people at that point in time when we got to the situation. And there was a lot of discussion, so we did see mutations, relevant mutations in the mink viruses. There was a lot of discussion, but they must be adapting, and that's less of a public health risk. Of course, you have this, there's tension in those discussions. I remember saying, we should not be taking this gamble because we just don't know. We don't know which effect those types of adaptations will have, and let's just not take the risk. It's hard to predict, and there's now in the network of people that study the variants and virus evolution. These are, of course, mutations that we're now also studying, comparing with let's say the UK variant B117 and the other variants to see which direction these types of mutations have, because I don't think we know that. But that was our primary concern. We don't want to take the risk that there is further evolution in a direction that we really do not want to see. And the second is indeed, establishment of a new additional animal reservoir. We talked about whether that would be possible in the mink themselves, but what I just mentioned is because it's such a wildfire on the mink farms, what we think is that it's not so likely because they're too susceptible. That this may, of course, be different if you have different breeds or if you have smaller farms. One concern was that so there was at some point something like 5 million animals that were all ready to give birth. So that is a factor 8 multiplication of animals in a very short period of time. So just virologically, you can just see this explode. So that's why we really decided to stop the sector.
Thank you.
Okay. Are there other questions here? Yes, Willing, please.
Hi, Professor Koopman. Thank you for the talk. I have a question regarding the mink infection. Can you comment on the clinical signs of the mink after infection and the tropism of the virus? Since ferrets actually have been experimentally infected, what are the level of similarity between minks and ferrets? Thank you.
So there was quite a mixed picture that we don't completely understand yet. So we have seen farms where there's a lot of disease and mortality, up to 8%. So that's quite substantial. So where the farmers actively picked up the phone and said, I have a problem. And we've had farms where the viruses were picked up through screening and there was very little disease. Now, this is, of course, over a period of a couple of months. What we've looked is there a difference between the clusters? Because the viruses differ somewhat. We cannot really see that. Is there a difference because of mutations? That's not something we've seen. Is there a difference because of other infections? So we are doing with a student from Hong Kong, Kirsty Kwok. So we're doing metagenomics now to see if there's maybe other co-infections that could explain part of the clinical expression. But so far, nothing clear there. So it is puzzling. So, but if there is clinical disease, then there is pneumonia, very very similar to what we see in ferrets. Of course, clinically, the ferrets are not that sick either. But what it does mean is you cannot expect this to pop up from just the disease monitoring. You really have to look whether or not the animals are infected.
And if I may, before closing, because I think we're a little bit running out of time, how can you monitor pathogenicity of viral isolates from these minks? Are you using, I don't know, ferrets in the lab, or what has been your approach?
So I think this is one of the tough questions also with the variants of concern that we're looking at. How do we determine this? I think we are struggling to answer those questions. So we've talked about this. Should we do comparative infection experiments? Ideally then, of course, in mink. But unless you have an almost black and white difference in virulence, which I don't really expect, you would really need very large scale infection experiments to look at any, tangible difference. You might need to look at tropism differences. So we're thinking about those studies, but it's not. I don't have a straightforward answer. I'm happy to be advised here, but there's a lot of expertise in your team around this.
Yeah, certainly not easy. You have a question? Okay, the very last one, and then we'll move on with the. Thank you. Yeah.
Thank you, Marion. That was a fantastic talk. I had two small questions. When you said that the remaining wild cats were still being fed, despite the minks all being culled, how wide do you cast the net to actually control the animals? And do you think that having those nests of these wild cats that could have sustained transmission was a risk? And also, there was a difference when you were looking at the environmental contamination with swipes and swabs, and the swipe seemed much more positive. So what's the difference in how you collect a swipe and a swab?
So swab and
Yeah. So
Yeah,
and
sorry, can you
swipes.
repeat?
Swab and swipes. Yes. How are you collecting swabs and swipes?
Yeah, so there's a protocol there. There's a paper out there that if you send me an email, I can send. And what's the first question?
that the nests of wild cats were still kept despite the other mink being culled, and that the wild cats were still being fed. It seemed really nice and charitable, but it seems like a risk for further spread.
I understand your question to say, did you not cull the wild cats?
Yes. Well, you know,
yeah.
or perhaps
Or-
a little bit less brutal, but yes.
Well, that's the word, right? So no, we did not, because the populations are small. So because we felt that they are not a likely candidate for an animal reservoir that sustains transmission. And because there are very strict rules about wild animals, even if they are semi-wild animals. So that's why we didn't do that. What we did do is then keep feeding them so that the infection could burn out also in the cats, because they would then stay put around the farms. So that was our attempt at containing this
Geographical spread.
Yeah.
Okay.
Yeah.
Okay. Well, there will be, I'm sure, other questions. Thank you very much again, Marion Coppens, for this initial keynote lecture. We can have an applause even from here. It's not virtual. It's a real one and very heartfelt. Thank you. Thank you for the talk. And now I'll
You're
pass
welcome.
over to Vijay Dhanasekaran for the panel discussion. Vijay.
Welcome everyone to the panel discussion for day one on virus ecology, emergence, and transmission. Today we actually have a wonderful expert panel, both online as well as in person. We have two international speakers who I'll just introduce in a bit, as well as three of our very own who would be sitting here on a panel. As part of this session, I'll be moderating this session along with Professor Tamara Giles-Wernick, who's online. I believe she's in Paris, and she's head of the Anthropology and Ecology of Disease Emergence Group. We're really looking forward to these presentations. So just to give you a brief overview of this panel discussion, so we're going to have these 5 speakers come and give presentations as well. So each presentation would be a brief presentation on their topics, followed by a longish panel discussion. And so we've organized the talks to cover a wide range of topics, starting from origins and diversity of viruses, mainly focusing on coronaviruses, but moving on very quickly to transmission mechanisms, and then control, as well as Malik finally would come and talk about what do we do next and how do we prevent future pandemics. For those who are online, feel free to participate in the discussion by posting your questions online, and then we'll try and gather as much thoughts from you as well and address those points within our panel discussion. So before I introduce the first speaker, I just want to run through the credentials of our panel, and I will invite the panel, the in-person panel, to come join me on the stage. Malik, Hoiling, and Nancy, please come and join me on the stage. So while they come and join me on stage, I'd like to just run through the credentials of our experts. Dr. Shi Zhengli is joining us online from China. And she's quite well known through the years now because she was included in the Time magazine's 100 Most Influential People of 2020. And her research has actually been under high scrutiny over the past 1 year. So we're very happy to actually have her on our panel. So Dr. Shi Zhengli is at the head of the Center for Emerging Infectious Diseases at Wuhan Institute of Virology in China. Her research focuses on SARS-like coronaviruses of bat origins. She studied at Wuhan University and Wuhan Institute of Virology, then earned a PhD at the University of Montpellier II in France. She returned to Wuhan Institute of Virology in 2002, initially focusing on shrimps and crab viruses. And then a turning point in her career came in 2005 when she published a major paper in Science with Peter Daszak and other researchers from China, Australia, and the United States, which reported the first evidence that bats harbored coronaviruses closely related to the lethal virus that jumped from civets to humans in 2003. In 2017, Dr. Shi and her colleagues, Koi Ji, discovered that the SARS coronavirus likely originated in a population of bats in a remote region in Yunnan. Our next panel member is Dr. Vyasna Duong, and he was a deputy head, and he's now the head since August 2020 of the virology unit in Institut-Pasteur du Cambodge. His main research work focuses on arboviruses and zoonotic viruses. In arboviruses, involves in both research and surveillance activities. He has particular interest in conducting research on emerging zoonotic viruses, including Nipah viruses, arenaviruses, and hantaviruses, and in detection of viruses circulating in wildlife, including rodents and bats. Dr. Hoiling Yen, sitting in the middle here, is an associate professor at the Division of Public Health Laboratory Sciences of the Hong Kong U Medicine School of Public Health. She received a PhD in epidemiological sciences from the University of Michigan, Ann Arbor, followed by a postdoctoral training at St. Jude Children's Research Hospital, Memphis, Tennessee. Her research interests focus on understanding the mechanisms of influenza transmission among and between different reservoirs, investigating potential virus-host interactions that affect viral pathogenicity and transmission, and examining the molecular determinants that confer antiviral resistance. A major research focus in our lab has been on transmission mechanisms. Ongoing research progress in our lab apply interdisciplinary research tools to study the modes of transmission for influenza and other respiratory viruses under laboratory settings and in the field, including at the animal-human interface. Her lab also uses genetic analyses, molecular biology, phenotypic measurements to study critical molecular determinants and their effects on protein functionality and virus fitness. She has ongoing collaborations with local researchers from University of Hong Kong, Guangdong Provincial CDC, Genomic Research Center, Academia Sinica, Taiwan, and St. Jude Children's Research Hospital. Dr. Yen is a full member of American Society of Virology and a committee member of iServe antiviral group. She serves as editorial boards in various scientific journals. We have Dr. Nancy Leung, a postdoctoral researcher in the Division of Epidemiology and Biostatistics at the School of Public Health. Her research focuses on infectious disease epidemiology with major interest in immunity to influenza and other respiratory infections. Her long-term research goals is to study the mechanisms of immunology in humans by combining the research fields of epidemiology and basic immunology. To this end, she has completed comprehensive trainings in the fields of epidemiology and basic immunology, studies of immunity, transmission, and burden of human respiratory virus infections. She completed an MPhil focusing on molecular mechanisms of antibody-dependent enhancement of SARS-CoV infection to evaluate the safety of a SARS vaccine candidate. And then a PhD with a series of field studies on the aerosol transmission of influenza virus, and postdoctoral research has been focusing on study design and data analyses of large longitudinal cohorts and vaccine trials on influenza burden, population immunity, vaccine immunogenicity, and correlates or mediators of protection in Hong Kong, mainland China, and the UK. Dr. Leung has also received scholarships supporting postdoctoral exchange at the University College London, analyzing data from the flu watch study. Dr. Leung has a strong interest in field epidemiology, in particular on designing and adopting electronic systems for data capture and quality control, specimen tracking, and workflow monitoring to facilitate the management of large-scale community studies. Finally, we have Professor Malik Pieris, is a clinical and public health virologist with a particular interest in emerging virus disease at the animal-human interface, including influenza, coronaviruses, and others. He's also a honorary director of our HKU-Pasteur research pole. His current research encompasses the pathogenesis, innate immune responses, transmission, ecology, and epidemiology of human and animal influenza viruses. In 2003, he played a key role in the discovery that a novel coronavirus was the cause of SARS, its diagnosis and pathogenesis. Currently, he's researching the recently emerged MERS coronavirus. He coordinates an 8-year multidisciplinary and multi-institutional areas of excellence program of the University Grants Committee of Hong Kong on the control of pandemic and interpandemic influenza. He's currently director of the Center of Influenza Research at Hong Kong University, which fosters multidisciplinary research with an integration of basic clinical and epidemiological research spanning the animal-human interface. He also co-directs the WHO H5 reference laboratory at Hong Kong University and serves on many standing committees and ad hoc advisory committees of the WHO and FAO. So with that, I'd like to invite Professor Shi Zhengli, who is going to present her first presentation. And her title for the presentation is Diversity and Origins of Coronaviruses, from SARS, MERS to COVID-19, Preparedness of the Next Coronavirus Disease. Dr. Zhengli?
Yes, hi, good afternoon and good morning to the people in Europe. Okay, I'm trying to share my slides. So can you hear me well? Can you see my slides? Yes. Okay. So today, I would like to thank the invitation to this discussion. Today, so I would not talk about the COVID-19. So I would like to continue to make a warning about the future prevention of the next pandemic or emerging infectious diseases caused by coronavirus. So I continue to warning it because based on the last 15 years, we worked on coronavirus, particularly beta coronavirus. So as we can know that in the last 2 decades, there are 3 coronavirus diseases emerged. So all these 3 coronavirus are believed to have animal origin and from bats and introduced to human population through intermediate host. Although there are some debates about the origin of SARS-2, I'm sorry, I made a mistake here. So the SARS-2, which are believed to probably originate in bats or pangolin, because there are some SARS-2 related coronavirus were discovered in pangolin. And 1 year after the COVID-19 outbreak, so are still unclear which animal involved in the transmission from the wild animal to human populations. In fact, after SARS outbreak, Chinese scientists are in the leading places, in leading position for the coronavirus discovery in China, particularly from the group in Hong Kong University led by Professor Yuan. In fact, among the new coronavirus species, more than 50% were discovered in China, and particularly from bats. So we made an analysis of all the coronavirus discovered in China, and as we can see that the high diversity of bats and also high diversity of bat coronavirus were discovered in South west of the country, like Yunnan province, and also the south province of China, like Guangxi and Guangdong and Hong Kong, this province. So if we look at the details of diversity, and we can see that there are some coronavirus species that we should put priority for the future study, and to prevent the possibility of the interspecies transmission from these coronavirus species. So the first one I would like to mention is the SARS-related coronavirus. So now we know that there are two strains from these species have jumped to human, caused respiratory diseases. So one is SARS coronavirus in 2002 and 2003, and another one is COVID-19, which occurred at the end of 2019. And the second one is the MERS cluster of coronavirus. So this is the cluster of coronavirus, including the species of MERS coronavirus and also HKU-5 and HKU-4. And even though there was no reported cases for the MERS coronavirus or the infection of HKU-4 and HKU-5 to human, but from the previous study, and the HKU-4 coronavirus have a high possibility to interspecies transmission in terms of the receptor usage, because this species of virus can use the same receptor as MERS coronavirus. And the third species of coronavirus, and we think is HKU-2 related coronavirus. So this species of virus have jumped to pig farm at the end of 26 in Guangdong province. Although this virus was not reported to infect human, but this virus has been demonstrated to have a very wide host range. So this virus can infect almost all the cell line from human, from primate cells, and also other mammal cells. So I think that this virus should be a high possibility to interspecies transmission if we don't prepare anything. And the fourth one is the HKU-9 virus. So this virus were only found in Yunnan province, southwest of China. So as we can see, all these three big cluster of virus, in fact, were discovered in southwest and south of China. So we think that if we predict the hotspot of the coronavirus infection in the future, and we think the hotspot should be in the southwest of China and also South China. So in the past 15 years, we focused on more SARS-related coronavirus after SARS outbreak. So we have discovered highly diversity of SARS-related coronavirus in China. So here we analyzed all the reported the sequence of SARS-related coronavirus, and we can see that this species of virus have a much wider geographic distribution. But if we look at the detail, and they can be divided into 4 evolution lineage. And there's a lineage 1 in which it's SARS coronavirus located, only found in southwest of China and also south of China in Guangdong and Hong Kong. And the lineage 2 and lineage 3 were discovered in middle and north province of China. And the lineage 4 only found in Yunnan province and close to the South Asia country. And then we use the represented strand to make a more clear phylogenetic tree, and we can see that the lineage 1 and the lineage 3 were grouped into a SARS-related coronavirus. So while this virus either discovered, most of them were discovered in bats. And the lineage 4 have a bigger, a separate group. So this linear flow is the SARS-2 coronavirus located in. So after SARS or COVID-19, in addition, the discovery of the similar sequence in bats, and there are also the sequence were discovered in pangolin. But all these samples were captured from China custom and from South Asia countries. So this is clear that the SARS-2 coronavirus will not be paid attention before the COVID-19 outbreak. And now we still have very few knowledge about how and when and where the COVID-19 coronavirus emerging. But based on the limited information, and we think that there is a highly likely the SARS-2 coronavirus emerging in the region of South Asia region, including China, Southwest region of China and Southwest Asia country. So in view of so many of coronavirus, and we would like to work on the basic virology to predict which kind of virus is more dangerous. So in the lab, we try to analyze the receptor usage. As we know that in the past 2 decades, the 3 coronavirus, 2 of them use the same receptor, ACE2 as a receptor. So in the past 15 years, we try to analyze the one which can use human ACE2 receptor. I think this group of virus should have the high risk than others. So we made analysis of the spike protein, particularly the region which corresponding to receptor binding with the human ACE2. So generally, all the SARS-related coronavirus based on the receptor binding domain can be divided into 2 clades. The clade 1, which has relatively similar size as the SARS coronavirus, and now we know also similar size as SARS-2 coronavirus. And the clade 2 coronavirus have a much wider distribution, but have some deletion in this part. And we did the analysis before, and we know that the clade 2 cannot use human ACE2 as a receptor, and we think that it's low risk. And the clade 1, we analyzed some of them from bat strain, and we know that this strain can use human ACE2 as a receptor, but with different binding affinity with human ACE2. So we predict that the sequence which the S protein RBD domain similar size with human SARS coronavirus, and this strain should have a high risk to interspecies transmission because they can use human ACE2 as a receptor. And later on, after COVID-19 outbreak, the other scientists also analyzed the SARS-2 related coronavirus, and they found some of them, particularly from pangolin. The coronavirus from pangolin can also use human ACE2 as a receptor, but the bat coronavirus have a low binding affinity. And another strategy is we would like to predict the risk. It's the animal experiment infection. So we try to isolate or use a reverse genetic technique to prepare the virus. And we would like to know if the bat SARS-related coronavirus can cause the lung damage in mice. So we use human ACE2 transgenic mice to test some of the bat coronavirus. And we know that some of them have a high pathogenicity than others. So we think in the future we should prepare some drug treatment or probably vaccine development against some highly priority virus as we predict. But in fact, what we worked in the lab is based on the virology. In fact, that's emerging of infectious diseases. There are many factors involved in, particularly in the interface between the wild animal and the domestic animal and the human population. So recently, there's some people analyzed the factor which play major role in the emerging infectious disease, and they found that more than 30% is related to human activity, including the land usage, and also because it's easier to understand. So the land usage make the wild animal interface between human and the wild animal much more close, and allow the human have a much more chance to contact the virus carried by wild animal. So I think in the future, if we wanted to stop the future emerging infectious diseases, we don't know which strain will jump, and we don't know when and where the emerging infectious disease will happen. I think that the best way is the surveillance, because with the development, with the discovery of many viruses, and we can design the diagnosis method and to do the extensive surveillance in the interface between wild animal and our human community. And this strategy, I think, is more effective to prevent the future emerging infectious disease. So I'll stop here. Thank you very much.
Dr. Shi Zhengli, thank you for your lovely talk. Maybe we can first take a couple of questions online and then move on to a broader range of panel discussion points. If there's any questions online. Oh, yeah.
So far, I've gotten, can you hear me there?
Yes.
Okay, yeah, thank you for a wonderful talk. So far, I haven't gotten any questions online, but I actually had a question just to sort of start off. I would say I'm a specialist in Africa and zoonotic disease emergence there, not in the part of the world in Southwest China that you're discussing. But I was wondering if you could say more about these land use changes, and specifically the kind of timing that you're looking at that would explain or how the emergence of these coronaviruses' increased risk would be facilitated by those land use changes.
You know, in the world, I think there's some region which have a much more rapid economic development, including China and South Asian country. So in this country, we have urbanization. So a lot of people live in the city, and also we need a lot of space for the people. So in fact, we have many deforestation in this deforestation, and make our human people much closer to the wild animal. This is one major reason. And the second, I know that the agriculture intensification. So we know that in the city we have no space for the agriculture. So now there are more and more farms were constructed in the remote rural area, and so in that area, the farms have a very limited biosafety control. So the farmer have a much more higher chance to interact with the wild animal. So during this interaction, and the virus maybe have much more chance to transmit it from a wild animal to human. So they have much chance than before. I think that this is the basic things is we have much more chance to contact the wild animal. And during this contact, then the virus have a much more chance to jump or either to the domestic animal and from domestic animal to human, or maybe jump directly from wild animal to human. But this is an ecological accident. So in the ecological accidents, I think that in my technique or knowledge, and we couldn't grasp every moment. But if we do the continuous surveillance, not only the virus discovery in wild animal, I think that we did a lot of work for the virus discovery in wild animal, but we did less work in the transmission chain. So in the future, I think that we did as much as work in the transmission chain, like the domestic animal surveillance, and also the human population, which exposed regularly to domestic animal or exposed to wild animal. I think that this is an important chain we should surveillance, monitor in the future.
Yes, I have a question. Hello, hi, Jean-Lee. I remember when you came, this is Roberto Bruzzone, when you came 2 years ago to the coronavirus virology course here at HKU-Pasteur, you showed slides in which you had taken blood samples, plasma samples from people living near bat caves that showed cross-reactivity with SARS coronavirus-related, antibodies. They had clearly experienced some contact, while actually people in Wuhan had none of that at that time. I wonder whether you have had the opportunity to extend your investigation in rural areas and find out whether there is more evidence of these initial species jumps, in those rural, remote populations.
Your question is if we do some surveillance in Wuhan?
No, more serological data in people living near bat caves in Yunnan. You showed some data already 2 years ago, but maybe you have more cases now. You have extended the geographical range of your observation.
In fact, the only well-designed work is the one I reported, or we already published. It's we do the surveillance, serological surveillance in a cave, in a villagers exposed in the cave. And after that, we didn't do the well-designed work, but we continue to collect some sample in the hospitals. So when, this collection is not well designed, so it's by chance. So if we have some sources or the doctors, they are willing to collect some sample, or the patient which visit the hospital are willing to provide their samples. And we collect some sample randomly, and we did some serological detection in Yunnan province. But till now, in addition to the one we detected some SARS-related coronavirus nucleic capsid protein antibody, and we have not discovered the more positive in the randomly selected sample. But we do found some serological cross-reaction of the nucleic capsid protein of the other different coronavirus, like HKU-10. But this randomly selected sample have a very low ratio of positive.
Hi, Jean-Lee. This is Malik. So thanks again for your talk. So you showed that when you were before COVID-19 emerged, you were risk assessing those bat viruses which bind ACE2 as high risk. And obviously, you were right in regard to SARS-2. But can we assume that coronaviruses that do not bind ACE2 are not at risk? Because after all, there could be other receptors in human respiratory epithelium that might serve as alternative receptors. Over?
So your question is the other coronavirus which don't use the ACE2 receptor, may use the other different receptor, have also risk, right?
Yes, that's right, yeah.
Yes, you are right. So we continue to look for the new receptor for the other different coronavirus. But till now, we have not found any receptor which can be used by the other SARS-related coronavirus. So this is how to say? Yeah, because we have a limited, we have a limited the people and a limited budget to work all of the coronavirus. So we only focus the one which already. We only focus on the one which phylogenetically close to the SARS coronavirus or MERS coronavirus. So for the other coronavirus, we don't know which receptor they use. We have not studied more. Yeah. So in the future, I think it's very important to screen all the to screen the receptor of all the coronavirus and to make sure and to predict which one is much more high risk. For example, the HKU-2 related coronavirus, and we did some receptor analysis, but we found this virus seems don't use a specific receptor because this virus can infect a very broad range of cell lines. So even though we don't know the specific receptor, but we think this virus should be dangerous in terms of cell tropism.
Hi, Professor Shi. I have a question regarding the SARS corona-like bat viruses. You mentioned there are 4 lineages that you found in China. Just wondering, based on the population immunity point of view, for future emergence, are those 4 clades or clusters, whether there's any cross-reactivity between them? Thank you.
I'm sorry, I can't hear very well this question. Can the moderator to repeat the question, please?
Sorry. So I will repeat the question. You mentioned there were 4 clusters or clades of the SARS corona-like bat viruses in China. Because of population immunity is a criteria for future pandemic emergence. My question is whether people infected with COVID-19 can be their serum will be cross-reactive with protective against these other clusters. Thank you.
Oh, I'm not very clear your question.
I don't know, I can try to re-explain that. So if you use those, say, strains, there are 4 clades of bat-related SARS coronaviruses. If you use, can you have any cross-reactivity? If you immunize mice with one clade that can infect mice, can then the sera from those immunized mice cross-react, recognize some of those other viruses that use other receptors and therefore cannot be easily tested in the laboratory? Is there any cross-reactivity between those 4 clades?
Okay, yeah. Your question is to ask if there's some cross neutralization between different clade, right?
Yes.
We only worked on the clade which use human ACE2 as a receptor. So even in this clade, the strain use the same receptor, but they have a low cross neutralization between different strains. For example, here I can show this picture. Here, for example, previously we worked on these two lineages. So the one lineage is much closer to human SARS coronavirus. And another lineage is SH014, which have a much more mutation compared to human SARS coronavirus. So if we do the cross neutralization assay, and we can see that this lineage is W1 can cross neutralize with human SARS coronavirus, but does not cross react with this lineage. Even though these two strains can use the same receptor. If I answer your question.
Yeah, thank you. We got two more questions, and then we should move on to the next speaker. Sophie from the audience here. Oh, she's done. Nancy?
Hello, Professor Shi. Thank you for your talk. So I have one question. I agree with you very much about we should do more surveillance on the transmission event, not just only on the virus evolutions in the bat. I wonder that relevant to this question is how do you select the geographical locations or specific populations of bat for the surveillance? Because do you take into considerations of the frequency of interactions between this wild animal population with humans? Because if you are thinking about spillovers, firstly, we would like to see whether there's actual, for example, physical interactions or at least in the environmental contaminations of those bat coronavirus, for example, and being transferred to the human. So is there any consideration of the human interactions when you select the site?
So your question is how to select the sampling to do the surveillance of the transmission of bat coronavirus?
Yes. Any consideration with the frequency of interactions with the human population nearby?
Yeah. For us, for whatever we, the human sampling is very challenging. Yeah, for I think for all the people, particularly because we belong to Chinese Academy of Sciences. And but even though we try to do that, so I think for us it's easier. It's firstly we try to locate the people which are highly exposed to bats. For example, when we published the surveillance in the human population, it's in the cave we found the SARS-related coronavirus. It's a big pool of SARS-related coronavirus, and the villagers have the house of the villager had about 2 kilometers. Oh no, not 2 kilometers. Much larger. It's around 10 kilometers from the villagers and to the cave. So we think that because the bats can migrate in this short distance. And I think that the best surveillance is look for the domestic animal and the people who, the farmer of the domestic animal, or the people who do the animal trade, and also the slaughtering. Yeah, all these people which exposed to the consuming chain or the transportation chain or the farming chain. So in this transmission chain, the people should be is a highly exposed populations. I think that if I design the experiment, I should choose these populations for the analysis.
Right, talking about risk of transmission or spillover, I think it's a good time to bring our next panel member to give a short talk. Viasna Duong is going to talk on the risk of virus spillover from bats and the example of Nipah and coronavirus. Viasna, take over.
I can't hear your question.
Sorry, Dr. Ji Zhengli. Dr. Viasna from Cambodia is going to give a talk. Please stay online and we can continue the discussion after his presentation.
Okay. So I can close my slides, right?
Yes, please.
Okay.
Good afternoon. Please wait a bit while I'm sharing my screen. Okay. Hope you see my screen, and I hope it's not the presenter one. I'm not sure if you see it well.
Yes.
Okay. Understand. Yes. First of all, thanks for inviting me to this very nice anniversary of the HKU-Pasteur poll. And I remember that I was once also invited to this anniversary. And thanks very much to Dr. Roberto for this invitation. And I would like to take this opportunity to provide an overview of the risk of emerging infectious disease in Southeast Asia with example of SARS-CoV-2 in bats and Nipah virus in Cambodia, and to a larger extent in Southeast Asia. So you might be familiar with this graph by William Karash. Just to highlight this, that viruses and also other pathogens continue constantly present and circulate in wild and domestic animals, and it's us humans who pose ourselves a risk when we try to further get into interface between these animals. And just a few examples here that how we so far encroach into animals. So land use change has mentioned several times already, how we live together, close together, how we treat animals, and also how we live and behave and put ourselves in contact with animals, which pose ourselves into a great risk in contacting and exposing animals present in those animals. So in this, I would like to show the first example of the SARS-CoV-2 related coronavirus in bat detected here in Cambodia and also recently in Thailand. So in Cambodia, we recently detected SARS-CoV-2 in 2 chamal horseshoe bats in our biobank of sample collected between 2010 to 2018. And the sample were collected, the sample positive for SARS-CoV-2 were collected in 2010 in the northern part of Cambodia, as you can see here. And at genomic level, the virus share more than 92% of identity with the human SARS-CoV-2. And just want to add here is that the new 2020 SARS-CoV-2 detected in Thailand, also in the same bat genus Rhinolophus, which is of species Acuminatus, sharing a bit more than 91% to the human SARS-CoV-2. And this discovery in the 2 countries, and also perhaps the virus might be present also in other countries in Southeast Asia, show that the ancestor of the human SARS-CoV-2 causing this current pandemic might present at a wider geographic distribution outside of China. And this discovery also provide an answer, but also at the same time give us many other questions, how this virus travels, move, and evolve across thousands of kilometers between Southeast Asia and Yunnan to emerge in Wuhan in 2019. And if we look at genomic level, and it's also well known that the coronavirus families are prone to recombination, and this is not a surprising effect. If we know that SARS coronavirus in general in high prevalence in bats, every time we sample bats and test for coronavirus, and we always find we are surely find coronavirus, and the way different bat species and general live together in close proximity in caves help the virus recombine through time. So as you may see here, our viruses recombine across the genome, including the spike protein domain. And if we look, I'm not sure how to move it. Okay. And if you look at the bat species carrying SARS-CoV-2 related viruses that we have known so far, it seems that the Rhinolophus bat species, different Rhinolophus bat species can carry these SARS-CoV-2 related virus, and the distribution area of these bat span from Yunnan to Southeast Asia and also into the Pacific. And if we see here the geographic distributions, we see also that there's an overlapping area between these bat species. And in our very small specimen analysis of the habitat network show us that these bats genetically are associated and are related, and it is also known that distinct bat species often share the same habitats and also sometimes co-roost and then share viruses between them. And another example is Nipah, and I know that SARS-CoV-2 is at the current stage, but we should not forget also other viruses with pandemic potential like Nipah, which is also found endemically in Asia. And we know that this virus caused big outbreak in bats, in pigs and also in humans in Asia until present recently in India, as we also know. And as in other countries, this Nipah virus presents in flying fox, and the virus detected in Cambodia in year 2000, but little is known about the impact of this virus in humans and animals in Cambodia. And in our recent study between 2013 and 2016, we have shown that about 1% of flying fox urine that we tested were positive by PCRs, and their serums, we detected antibodies against Nipah in about almost 11% of blood sample we collected from bats. Fortunately, people that we sample in the area, living in the area in close proximity to this flying fox roost, as you may see here. This is the two areas that we study, but there are also other flying fox roost present across the country, and people living in these two locations and also other living at the area where these flying fox frequents for foraging did not have any antibodies against this virus, showing that perhaps there is no previous infections exposure to this virus. However, our study showed that human practices in Cambodia pose people a lot at risk at exposure to the bats, in contact with bat and exposure to Nipah, like consumption of raw palm nectar, hunting, consuming of bats, and in terms of knowledge, they have very low perception of the potential risk that bat might pose to them. When we look at the diversity of the virus, Nipah viruses that we detected in Cambodia, here we show in this phylogenetic tree in red, and the blue one virus is detected in Thailand, showing that there is a large diversity of this virus in flying fox, both in Cambodia and in Thailand. And the virus detected in both country cluster, in both Bangladesh cluster and also in Malaysian cluster. Between the virus in Cambodia and Thailand, the virus are almost the same, more than 99% of identity at nucleotide level. And in a recent study by a French group in Lyon, using one of the virus isolate detected in Cambodia, in Pteropus lilae, in P4 there, they have shown that the virus from Cambodia, here present at 381, has a similar or even high, even though it's not significant, able to infect a different cell line. Here is a human pulmonary cell line. Here is the alveoli cell line, and here is the bat cell line. And they also did an animal experiment in hamster. The blue line one is the Malaysian, and the red one is Cambodian. And they showed that the Cambodian virus is as virulent as the Malaysian isolate that caused outbreak in humans and also in pig in 1999. It's a bit slow, it's stuck, it doesn't move, sorry. I'm really sorry, the PowerPoint just crashed. I need to reopen it again. I'm not sure if you still see my screen.
Yes, please go ahead, VS now.
Okay. So here just an example of how people here in Cambodia live in close proximity at interface between wildlife and animals, domestic animal. Here an example in Battambang province, in the northwestern part of the country. Here present here a morning market where people coming into this market to buy fruits, vegetable, meat. And just above them in a big tree, you see this black dot is 100,000 of flying fox hanging over their head, dropping urines and feces on potentially on them and also on those vegetable and fruit selling underneath. And here I had the opportunity to take a few photos of bat, because these bats do not stay still and they do play and together, and sometimes they just fly away. And if you can see well, if you notice here in the circle, that during their flight they do urinate while flying, and this urinate can drop on any animals or persons underneath. So in terms of risk emergence in Southeast Asia, as we all know that in that region is a hotspot of emerging infectious disease because due to different factors. But so the risk is present here. The virus with pandemic potential are present: Nipah viruses, SARS-CoV-2, avian influenza. And also there is an interface between humans and animals, risk behavior, low perception of risk, among populations. And this and what is really missing for the spillover in this region. And perhaps we just all these are just hypotheses, and we need would lead to further questions and also investigations. Amplification effect. Do we need an amplifying cause? Intensive farming. But is there any ecological barriers in these interfaces? Or whether there is already an unrecognized sporadic outbreak, but we don't have any diagnostics tool, or there is a lack of surveillance to prove and to document these small outbreaks. The prevalence and survival of the pathogens in environment where it is hot and dry, we still don't know. And whether there is really a need, a bad luck or chance when there is a right viral candidate, right time and at the right place. And this on the right-hand side chart just really presents very well what is the chance of a spillover events from animals to humans. So we need all these different layers of factors to align together in order for the virus to emerge in human populations. And then what should we do to prepare ourselves? And to my own opinion is to have kind of a long-term longitudinal surveillance, which is, I think, crucial to be prepared for the next pandemics, not in specific country, but at a regional level, focusing on One Health approach, surveillance to monitor potential emergence. And also this surveillance should be conducted at interface between humans, wildlife, and also domestic animals. Regular sampling to understand the seasonality of virus shedding. Through our study here in Cambodia and also in Thailand and also other part of the world, we know that the virus are shedded in from their reservoir, for example, bats, at very specific time of the year. For example, during the time when there are a lot of juvenile, which represent susceptible population with low immunity, or during pregnancy, lactation, where the bats are stressed and at the same time they shed more virus. So understand all this make us more let us have maximize our probability to detect and also to provide effective intervention measure to prevent any spillover event. And also, more importantly, besides documenting all this, but what is important is to the behavior change and understand of this perception of risk, and also how to find a way to provide a long-term change and a sustainable change of this risk behavior, and how to try to curb cultural traditional habit. With that, I would like to thank for your attention, and also to thank all different partners that help in all this work. And thank you.
Thank you, Dr. Vyasna. Just because we're running quite into the next three talks, I just want to raise one point, because both the speakers who spoke about the origins talked about the human impact. Dr. Shi Zhengli mentioned ecological accident, and Vyasna mentioned human practices and risks as one of the major factors for emergence. I was actually wondering, is there a better way that we can, address, quantify such behavior? For instance, in China, we've had a massive reduction in pork production due to African swine fever over the last 2-3 years. And I was wondering if people could comment on how such practices could impact access to these bats, especially with coronaviruses as a focus, because I think coronaviruses and Nipah are completely different in terms of the ecology and spread. So focusing on coronavirus prevention, preventing future pandemics. So how does food consumption and other practices, and if that's something that we can quantify in your field research?
Yeah, I'm sorry if I do not understand your question. I barely hear your voice, but I want to repeat what I understood is that how we quantify the risk, right, about coronavirus spillover to humans here in Cambodia. Is it correct? And what are the interface?
Yeah, sorry. I mean, you mentioned human practices, so that's one of the main reasons driving emergence and risk of infection. So I was wondering if there were long-term or short-term changes in human behavior. I mean, of course, human population is increasing, so the population size increase is obviously going to increase risk of infection. But I was wondering in terms of other factors, availability of food, famine, African swine fever, which has a massive impact on pork production recently, which has had an impact on food consumption in this region. So does your research provide any clues or indication of how we can address these factors directly?
Yeah, so different factors are present in different interface, but sometimes there is a missing part of the puzzle. For example, we have here in Cambodia and also in Thailand, Vietnam, and Laos, villages where people raise bats for guano, because bat guano is precious and also considered the best fertilizer. So people rear bats just at household setting, just in front of their house. So, and then people walk and animals also walk wandering just underneath these bat roots. And in the evening, this bat fly out of the roots and or at the same time urinate. And I was there, and it's just like a light rain when we walk under when they fly. But we study the presence of coronavirus there in those flying bat farms, and we do detect coronavirus, but all of them belong to alpha coronavirus and cluster very closely with other bats, not close to any human coronaviruses. So the exposure there and very intense, but the potential virus to transmit to humans is not. It might take time, more time, and we don't know how long the virus recombine and mutate to be able to infect humans. But until presence, we do collect sample from people living in those areas. We do not detect any coronavirus antibodies. And while the other example is SARS-CoV-2 in bats that we detected in the Chamelai bats are located in very remote area and with difficult access to that area. So the potential virus with emergence potential is there, and there is a close proximity with other intermediate hosts present in those cave and karst area, but contact with human is quite rare. And the third example that I provided you is about Nipah, and the virus have the potential to infect humans. Interface is there, but there's no transmission. There's no report in humans. So what is the missing puzzle? We still don't know. More investigation, more work need to be done.
Thank you. If there's any more questions or comments for Viesno or any other comments on this same subject area?
Thank you. Another really great talk. I'm just interested, are you supportive then of the Global Virome project, which is interested in looking at repeated virus isolation at these crossover sites? Or do you think that it's more important to have that early, rapid sequencing and isolation of viruses that have crossed over rather than trying to do a risk assessment on these unlimited viral threats? Does this make sense? So either that you have the global virome surveillance of all the viruses that really are out there, or if you're just looking for the spillover events that have the human cases occurring very quickly being identified.
Vyasna, if you haven't spoken, this is really a question. I think it's an interesting point that anybody can comment on. Do we keep looking for viruses in bats and other animals, like the virome projects conducted globally? Or do we have to enhance surveillance in humans or interfaces between animals and humans so that we can actually identify the ones which matter the most?
I'm not sure if I understood well the questions, but I do think I forgot to mention that about the virome as well, is that we do use, for our current surveillance, we do use a lot of labor-intensive work, technique assays, like conventional PCRs at the family level, trying to detect known and new viruses, but this is very labor-intensive and take a lot of time. And the new technology, like NGS and also this pipeline for virome and microbiome or virome in the bats and rodents would help a lot us to understand the diversity of these different virus species in those wild animals.
Thanks, Vyasna. Malik?
Yes, so I think there's a lot of enthusiasm for more and more sequencing, which, of course, I'm sure, Vijay, you will be very happy with. But I think we have to realize, I mean, if we look, we will find, and we'll find hundreds of thousands of viruses. So how do we risk assess among them? And I think that is indeed something I will touch upon in my talk briefly. So I think scanning the horizon is important, but finding ways to risk assess them is probably even more important.
Right.
Yes, just comments that I do understand the questions. Is that I think it's important to have an inventory or a diversity of bat, but on the other side, it's also important to identify potential candidate of the virus, and also to have human samples that we might be able to go back if we find a potential candidate. And there's a tools that we collaborate with IP Paris, is that with the genome sequence that we have, they can synthesize the peptide specific to the sequence we detect, and then use this as serological tools to investigate humans' exposure to this candidate new virus. This is very powerful tools that we can help us understand better the risk.
Thank you, Vyasna. I'd like to encourage you to stay online and participate in the discussion follows. And now I'd actually like to move on topics to transmission mechanisms, and I'd like to invite Hoi Ling to give a presentation.
So I will briefly go through the transmission mechanisms, and we'll be focusing on SARS coronavirus 2 and influenza viruses. So influenza viruses predominantly replicates at the respiratory tract, while the SARS coronavirus 2 replicates at the broader tropism. However, for both viruses, the major route of emission is likely through the aerosols that's expired through either speaking, coughing, or breathing. So this image just shows you while a person is sneezing and a cloud of aerosol is being generated. So in this talk, I will be referring to aerosols as airborne particles without a defined size range. So from human, we know that from healthy subjects, so this is a study done with healthy subjects, so while they are speaking, coughing, or breathing, there's a wide range of aerosols that's being produced, but majority of them actually are below 20 micron. So with this in mind, if we have a scenario like this, and subject A is infected with either influenza or SARS coronavirus 2, this subject is going to release a cloud of aerosols of various sizes. And person B is a susceptible host in close proximity to subject A, will be exposed to various sizes of particles. So this has some implications. The size distribution of the particles has implications for the temporal and spatial distribution of those aerosols subsequently. So for scenario here, for subject B, he's going to be exposed by multiple routes, including the large droplets that may be directly deposited on the mucosal surfaces, or inhaling fine aerosols into the respiratory tract. Or some of the large fomites will deposit in the surfaces surrounding subject B, and that person can also be exposed to fomites that's deposited. But in a longer distance, we have another host, subject C. In this scenario, transmission can be mediated by fine aerosols only because of the ability to travel longer distance. So in order to study the multi-route transmission potential, we used the golden hamster as a model. So we started by looking at the contact transmission potential, inoculating a donor, and then starting from day 1 to day 14, we introduced a naive contact to co-house in the same cage with the donor. So with this scenario, the contact ferret, the naive contact, is going to be exposed by multiple routes, right? So droplets, aerosols, fomites, all of them. So we found that transmission in this setting is very efficient, 100% in 3 out of 3 repeats. We then continue to see what is the communicable period of the infected donor. So we changed the setting slightly. So instead of introducing the naive animal on day 1, we introduced the animal on day 6, post-inoculation of the donor, and continue to co-house them again for 14 days. And under this setting, there is no transmission observed, so suggesting that the communicable period was very short. We then move on to test the aerosol transmission potential. So in this experimental setting, we have inoculated donor, and then the aerosol contact was introduced on day 1 when the donor was infected, and only exposed for 8 hours in the iso cage with a setting as shown here. We found that it is actually very efficient, so only exposing for 8 hours of the aerosols, and all of the contacts become infected. We also tested the fomite transmission potential in the hamsters. So to do this, we have inoculated a donor and housed it in a cage for 2 days. So it becomes a soil cage. We then move out the donor and move in a naive hamster in the same cage, and again house for another 48 hours. With this experimental design, we found that transmission was less efficient. Only 1 out of 3 repeats, we can see transmission. So with this setting, we can conclude that the dominant route of transmission in hamster was mediated by the aerosols. So because we mentioned that there's a wide range of aerosols being generated, so we were quite interested to understand what is the transmission potential by different size fractions. So here, this is a study done with influenza virus, not SARS coronavirus 2. So with influenza, we know ferret is a very good model, and they similarly can transmit efficiently by aerosols. So we know that they emit different sizes, and then we use this device called an impactor, trying to separate the large and fine aerosols by size. So the design of this setting is that you have airflow coming, and if you have very large particles, they will be impacted on this impaction plate because of this inertial impaction. But the fine particles will be able to travel with airflow to the next stage. So using different sizes of the impactor, we can control the cutoff values and control the particle sizes that's being passed through. And we have this experimental setting. We have donor ferrets on one side. They are emitting aerosols of various sizes, and we have an impactor installed in the middle. And on the other side, we have naive animals being exposed to the specific aerosols by size. So using influenza virus H1N1 and H3N2, we found that with different sizes of aerosols, you start to see, with the size going down, you start to see decreased transmissibility. Until when we use the 1 micron impactor, and then we see no transmission for both viruses. So the 1 micron impactor actually separates, so that has a collection efficiency D50 of 1 micron. That means 50% of the particles at 1 micron will be deposited at the impaction plate, whereas the other 50% will go through. The same impactor, on the other hand, will have a D95 at 1.5 micron. That means 95% of particles at 1.5 micron will be impacted on the impaction plate. Therefore, using this, we can say that transmissibility actually are mediated by aerosol larger than 1.5 micron. But interestingly, we found that similar like humans, majority of the exhaled particles are actually less than 1.5 micron. So that's very interesting. So only about 23% of the exhaled particles actually containing transmissible viruses. So we also were interested in where do the transmissible viruses were expired from. So one possibility is that they were formed lower in the lung through the breathing mechanisms. Particularly the exhalation has been proposed at the bronchial. You will be able to release a large quantity of fine aerosols that way. Or alternatively, can be viral replication at the upper respiratory tract. So to address that question, we inoculated ferrets either intranasally or intratracheally, so that they have the viruses replicated at specific anatomic site. And then we monitor their onward aerosol transmission potential. So to do this experiment, we have used a pair of viruses. This is the H1N1 pandemic virus. They are the wild type, and then a barcoded one. So the barcoded one has amino acid sequence completely identical to the wild type virus. But at each of the gene segment, there is a barcode sequence that's separated by about 30 nucleotides. So we inoculate in this scenario wild type virus to the intranasal route, whereas the barcoded virus to the lung. And then the donor was allowed to donate the breast to the recipient on day 2 post-infection for another 8 hours. So this is a short time point exposure. We then monitor the viruses that's being shed by both the donor virus and the contact ferret and the donor ferret. And then next generation sequencing was performed to check the genotype that's being transmitted. So from the donor, we can see that they were predominantly shedding the wild-type virus that's inoculated in the intranasal route, not the barcoded virus, which is in the lower lung. And it is also the wild-type virus in the upper respiratory tract that is also transmitted to the contact animal. Even though later time points, we can see there was one sample showing increased barcoded virus in the nasal sample on day 5. So we also performed the experiment in reverse. So by inoculating the barcoded virus intranasally and the wild-type virus in the lung, and then also confirming that in this scenario, we can only see the barcoded viruses replicating in the donor, and it was also transmitted to the recipient ferrets as well. So what we have learned so far are SARS coronavirus 2 and influenza viruses are transmitted by multi-routes, and short-range aerosol transmission may be the most efficient mode. A majority of the expired aerosols in humans or in animals, so far we've seen in ferrets, are below 20 micron. Influenza viruses are transmitted between ferrets by aerosols larger than 1.5 micron, and transmissible aerosols will expire from the upper respiratory epithelial cells. But there are many things that remains unknown. Effect of environmental parameters on infectivity of expired aerosols. So how do we explain for the winter epidemics? mechanisms facilitating aerosol formation from the upper respiratory tract, and maybe different vocal activities, speaking, singing, and we know there's definitely some super emitters that have been reported for the SARS coronavirus 2. And finally, the effect of pharmaceutical interventions targeting upper respiratory epithelia, can those prevent onward transmission? So finally, I'd like to acknowledge the funding and the students and the collaborators who have contributed to the study. Thanks.
I'll just wait a second till the panel's online. I'd like to quickly check with my co-moderator, Tamara. Is there any questions online?
No questions online yet, but I think you should circle back because it seems like the questions are sort of generated by also what's going on in the audience too. People need time to think.
Okay. Right, there's a couple of questions in the back. KG?
Thank you. Hui-Ling, one of the really practical questions is how long is a person infectious for when they have COVID infection? And so one of the surrogate ways of trying to get that is to look at CT values and so on when you're doing PCR. But it's essentially a surrogate approach. And so your experimental data genuinely measures infectivity, the period of infectivity. So are there any human studies which relate to that? Because it has a huge impact, I think, on quarantine, how to actually handle a pandemic like this and handle individuals.
Yeah, so we also measure the viral RNA in the nasal washes, and from the infected hamsters, they can continue to shed pretty high copies of RNAs in the nasal washes for quite some time. Actually, we've monitored for about 30-something days. They can still have RT-PCR positive samples being taken after infection. So
if they're shedding high amounts of RNA, but they're no longer infectious,
they are no longer transmissible.
What's going on? What do you think is going on?
Yeah, so we have actually looked at patients, indeed, in response to the hospital authority, because they were worried about locking up patients for almost a month because they continue to be PCR positive. So we looked at culturable virus, whether you can culture the virus, PCR, viral load, but whether you can culture the virus, and also Leo looked at subgenomic RNA, which is a marker of replicative virus. And the bottom line is that after day 8, really most of the culturable virus is by day 5, day 6, very similar to the hamsters, but the outside limit is about day 8. After that, you cannot culture virus. Although PCR can be positive at quite high viral load for a long time, just like the hamsters. So actually, the two models are very, very comparable. And indeed, there have been a number of other studies now, a large study from the UK, which has confirmed that. Basically, day 8 or so is about the limit. There are big epi studies looking at contact tracing, which have also confirmed that there is no evidence of, or very little evidence of transmission beyond day 5, day 6, day 7. Now, all this, of course, applies to mild, asymptomatic, mild, or moderate cases. Severely ill patients, or immunocompromised patients, clearly can be infectious for a much longer period of time. But I think the evidence is quite clear. Leaving aside the severe and immunocompromised, for the moderate, mild cases, you can happily discharge them by day 10, even if they are PCR positive.
Yes, have you did some quantitative measurement of how many viral particles or PFUs are released per minute, per hour by the animal or by the patient?
You mean for the SARS-2 virus or the?
For the SARS-2.
We are in the process of doing additional study trying to measure that in the breath of the hamster. So this is still ongoing. Yeah. Thank you.
Just quickly checking with Tamara. Is there any questions online?
Yes, there's a question from Janice. Does the speed of partial projector producing, I think she means particles or partials, affect the study? How do you monitor or adjust the effects if there is any?
Could you just repeat that question again, please?
Yes, from Janice, does the speed of partial projector producing, it says partials, but I think maybe it means she means particle, or it might be a typo. Particles affect the study. How do you monitor or adjust the effects if there is any?
So for the study with the SARS coronavirus 2 in hamsters, we are not able to conclude the size range, that mediates the transmission by the aerosol route. So indeed, we know that the fine aerosols are able to travel further. But then at the same time, if it is a large droplets with a high velocity coming out from the mouth or the breath, actually that particle can also travel pretty long, even though it's a large droplets. But with the impactor usage, on the other hand, we can control the size despite of the velocity that was coming out from emitted by the animal and so on and so forth. Yeah. So hopefully that addressed the question.
Right. Thank you, Hoiling. I think we just, due to lack of time, I think we just move on to the next speaker. Dr. Nancy Leung is going to talk on controlling the transmission of respiratory viruses.
Thank you for the organizers for inviting me back. Actually, I'm an alumnus of the HKU-Pasteur Center, so I'm honored to be invited back to give this talk. So I'm an epidemiologist, that's why I'm going to talk more about the respiratory virus transmission in the human. But I think that by looking at those transmission, there may be some insight or some interesting questions that can raise and to inform research for identifying virus emergence. So in particular for this talk, actually it will be mostly based on a review that I recently written for the Nature Review Microbiology, which is going to be out next month. And that reviews mostly about the transmissibility, transmission, and the control of respiratory viruses. So today I'm going to focus on the control part, but there's an interesting discussion on the aerosol transmission of the SARS-CoV-2 if you're interested. So basically just to have a brief introduction of the human respiratory viruses. So we define those viruses as viruses that can infect cells of the respiratory tract that could elicit respiratory symptoms, but may as well other symptoms, for example, gastrointestinal symptoms as well. And we believe those viruses can mainly transmit by respiratory secretions of infected persons. So as I will show in subsequent tables, actually they come from very diverse virus family, and they differ a lot in their viral and genomic structures, population susceptible to the infections, severity, seasonality, transmissibility, and modes of transmission. And I'm going to talk a bit about on the transmissibility and modes of transmission. So if we look at all the respiratory viruses in the human that is commonly known, so apart from the coronaviruses and influenza viruses that has been discussed in the earlier talks, people were also familiar with other viruses such as the measles, which is highly transmissible, RSV infections in the children, chickenpox, which is the VZV, and also rhinoviruses causing the common cold. So for these viruses, you see the coronaviruses and the flu viruses, we already know there are zoonotic viruses in the animal populations, in bat, in avian, etc. And some other human respiratory viruses, I'm not so sure whether there's the equivalent zoonotic viruses in the animal populations. We would also look at how transmissible they are in the humans, and there are several different types of measures that epidemiologists use to look at them. One of them is the basic reproductive number. So if we focus on the basic reproductive number, which in short R0, we define it as the average number of successful transmission per infectious individual in a population, meaning that for each one of the infectious individual, how many number of people will be transmitted by that person. So when we know that there's one infectious individual transmitting more than one person, then there will be sustained human-to-human transmission. And that's how we assess transmissibility of the viruses in the population. So for each of the viruses, if we look at the R0 and for comparison, for example, for flu, where we see there's a zoonotic viruses and there's endemic flu viruses. For zoonotic H7N9, we see that the transmissibility is less than 1. That's why we expect there's no sustained human-to-human transmission. And for the endemic flu viruses, for example, the flu H1N1 emerged in the 2009, we see that the R0 is slightly above 1 to some study report 21 as well. So that's why it becomes a pandemic and having transmission in the populations and now become endemic viruses. So one of the questions we ask is what makes the difference between these two types of the same flu viruses? Was the determinants for the emergence of pandemic potential? So for the coronaviruses, so there are two spillover or eventually becoming pandemic for SARS-CoV-2. There's two SARS-CoV related viruses. The first one is the one emerged in 2003. So at that time, actually, we estimated the transmissibility can be more than 1, can be as low as 3, while for the current SARS-CoV-2 emerged in the last year can be as most as 6. So in some way, actually, just by looking at the transmissibility, you would expect that maybe SARS-CoV-1, someone would call SARS-CoV-1, would expect to become a global pandemic if we just look at the transmissibility estimate. But actually, one of the things that we also consider is when will the individuals be infectious. For example, now we most believe that the SARS-CoV-1 will only be most infectious after symptoms onset, while the SARS-CoV-2, there are a lot of pre-symptomatic transmission. And we believe that that's one of the reasons why SARS-CoV-2 can become a global pandemic, while SARS-CoV-1, because of this nature, we can do a proper control at the population level to prevent it to become a larger pandemic. So stepping back, just to compare in general different viruses, I mentioned before, they are highly transmissible respiratory viruses like measles. With some, there are a lot of different estimates, very heterogeneous, but some can be as much as 700 or above. And also there's a rhinovirus where we expect there are a lot of people with repeated infections in their lifetime with rhinoviruses. So they're endemic in nature, but actually the transmissibility is not very high. So why there be some viruses with such a high transmissibility while some viruses are lower transmissibility in the human population? So as Herlin has explained very clearly, there are different modes of transmission, including the direct contact, indirect contact, droplet, and aerosol. So there will be some hypotheses where each of the different modes of transmission will contribute differently to the transmissibility in general. For example, whether some viruses will be more biased to a certain mode of transmission and whether it will lead to an increase in transmissibility. So if we want to look at the individual evidence to support each individual modes of transmission for all the different respiratory viruses, you see that actually for each of us, for example, the coronavirus, there are already some evidence, not only about SARS-CoV-2, but also there's some endemic seasonal coronaviruses that has been circulating in the past 50 years. So among all these coronaviruses, we see there are evidence in support of direct contact, including a transmission of the infections via hands in observational study. And also there will be evidence in support of the fomite route where you discover infectious viruses on naturally contaminated surface or transmission. Not much study in droplet, and quite a lot of study actually on the aerosols, and similar for the influenza viruses. So actually for these viruses, they have different types of or levels of evidence for individual modes of transmission. And especially that when we are very concerned about aerosol transmission, so for the two, at least for the two viruses that people mostly believe they transmit for aerosol route, the evidence coming from like there's a transmission of lab-confirmed infections by aerosols in frontier study. And that's why actually in a lot of the design of interventions to prevent transmission, for example, in healthcare settings, as recommended by the WHO, these precautions are modes of transmission targeted. So you see that actually for the measles virus and also for the chickenpox viruses, airborne precautions in healthcare settings is recommended because of this evidence of lab-confirmed infections in humans. So recently, we published a paper about observing large URTI upper respiratory outbreak in Hong Kong after school reopened, after prolonged closure due to interventions for the COVID. So the interesting observations that we had is that during the last year in Hong Kong, we have quite a long period of school closures. So we expect that this type of very drastic measure will prevent transmission of all respiratory viruses during a closure just because there's no contact between the children, etc. Basically, very drastic social distance measure. But when we reopen the school, actually, we also implement some individual level of interventions such as face masks or spacing between desks, etc. And the interesting fact is that actually we see a larger number of outbreak and a larger size of outbreak after school reopening in school children. And if you compare this data with the data in previous year, it's very big. And then more interestingly, actually, if you look at the etiology of this outbreak, you don't really see SARS-CoV-2 transmission or the flu transmission, but rather they are mostly from the rhinoviruses. So in some way, what we are saying is that for the same interventions or population-level interventions that we used, actually there could be different effectiveness on virus transmission for different respiratory viruses, at least in the population level. And one may ask, why is it like that? So I mentioned that there are different levels of evidence for modes of transmission, and for each viruses, they may transmit with different relative contribution with different modes. So for the MPI, standing for the non-pharmaceutical interventions, for the MPI we use actually each MPI would be specific to a certain mode of transmission. For example, the hand hygiene would be specific to the physical contact, the face covering would be more for the droplet and aerosol. And because of this different specific modes of transmission being intervened, so maybe that's one of the reasons why the interventions will work better than other on one viruses than other viruses, just because viruses transmit differently. Moreover, even for the same route, and the interventions may have varying effect on different viruses. For example, there are data suggesting that alcohol will be more virucidal on amphilovirus than non-amphilovirus, and that is quite often the case in terms of virus inactivations through different means, that the non-amphilovirus is actually more persistent in the environment. And also, one thing that I think we need to be careful about is that how do we measure effectiveness of interventions? So mostly we will see data suggesting there will be mechanistic evidence in support of the virus inactivation or reductions, but whether it translated to reduction in the transmission is not necessarily the case. For example, for hand hygiene, although we observe there's significantly reductions associated with reduce in respiratory illnesses, but actually if you look at using laboratory-confirmed flu infections as an outcome, we do not see significant reduction. This could be artifacts in the study design, insufficient sample size, insufficient compliance, but nonetheless, sometimes when you see in the media people quoting there's no evidence in terms of reduction in transmission, actually there'll be some evidence in terms of virus inactivations or reductions. So it also similarly for face covering and also for other systematic levels of MPI measure, such as surface cleaning, ventilations, and using ultraviolet irradiation as a desanitization of the air. So there are existing data in the literature showing that each of the interventions actually will have different effect on different mode or on different viruses. So it brings to my last slide is that when we're talking about controlling virus emergence, I, as an epidemiologist, would want to ask the virus like this question. For example, what would be the potential of the emergence for zoonotic viruses of other virus families? So as we've seen, we see coronavirus and flu virus that emerge, but how about all the other virus family? Would there be potential for emergence as well, especially for virus family, which we already know there will be highly transmissible virus, including the measles and the chickenpox? Also, because when we look at pandemic potential, we are not only talking about just transmissibility of the virus itself, it also depends on the period of infectiousness and its relevance or in relationship with the symptom onset. So will there be like a study or a need to assess the pre-symptomatic transmission potential in emerging viruses? Because that's one of the determinants where they become global pandemic. And of course, there will be like user questions about what's the viral determinants of preferential mode of transmission for each of the viruses. And also, I think there will be less study on that, where there's a viral determinants to indicate the effectiveness of intervention specific to a mode, specific to our viruses. So that's what I want to share, and I welcome your questions. Thank you.
Thank you for the wonderful presentation, Nancy. I'd like to just quickly start off by talking about masks, because I think Hong Kong was at the forefront in actually explaining globally why they're so important. Could you just comment on some of your work that you've done and how it's explained in identifying the role of masks in controlling the transmission of respiratory viruses?
Features referring of one of our publication last year talking about the effectiveness of face masks in terms of source control. So actually, before this paper, there will be some existing study on the effectiveness of face masks in terms of source control, mostly on flu viruses. And also, there are some study in terms of the infectiousness of influenza viruses in aerosol. So in some way, I think for many of us, we already know that face masks will work as a source control. So what is the added knowledge based on that study is actually expanded to other viruses. So we look at coronaviruses, we look at rhinoviruses as well. As I've mentioned, they cause a different spectrum of disease in different populations. And most interesting thing is that we realized there will be difference in terms of the effectiveness for different viruses. So if based on the study I mentioned just now about the outbreak, is that if we think that one interventions will be effective for all respiratory viruses, maybe this is not the case. And I think we have to be really careful about using measures we traditionally believe will be useful on new viruses that emerge. There will be also other heated discussion about using face masks as a protection for healthy individuals. And I think that one thing that people haven't realized is that the further away you are from the infected individuals, of course, there will be a decrease in the virus, in the risk of infections from that infectious persons. So when people have been asking about why there's no data in assessing the protections of the face mask, but actually somehow the risk level will be so low that you will not be able to see the protection just because there's no risk in the first place. So personally, I would say that the current recommendations of mandatory masking in a lot of population is more thinking about on the source control, and that's why there's the need using it like for everyone instead of those who are symptomatic, etc. So, but I just want people to realize that it could be useful and have to look at the different types of evidence and be careful on what each type of evidence infer.
Thank you, Nancy. Is there any questions for Nancy or any general comments? Just checking with Tamara online. I don't think so. I think.
What about Nancy? Have you included in sort of these summaries as well the face shields? That apparently there's a lot of you can have a lot of infection through the eye route as well, and that it's really important to cover that for SARS-CoV-2 as well. What do you recommend?
Yeah, so there will be some small number of studies showing there's auricular, like infections through the auricular exposure of the eyes, like there are 1 or 2 studies on flu. But actually, there's not much study, I would say, looking at how useful is face shield in terms of the protection, etc. Just because there's not much research. But I want to raise the point that face shield originally it's used in healthcare setting, and that is to prevent kind of like a larger droplet or spillage in the hospital settings when they are doing procedures. So I think we have to realize that some interventions will be more useful in healthcare settings where there will be higher risk of infection through all different types of procedure. But when we directly adopt it from the healthcare setting, high-risk settings, to community settings, it may not as useful as in healthcare setting, just because the risk is a much lower.
Is there any more discussion? I'd like to invite Professor Malik Pires, who's going to present a short talk on pandemics. Will we be better prepared next time?
Thank you. So I know that all of you are really concerned about the pandemic now, and you're probably not thinking much about the next time. But I think this is the best time to think about the possibility of the next time and the lessons that we have learned from this time, how we can apply that. So the impact of this pandemic, I don't have to tell you. All of you have been impacted severely. You know the numbers, 110 million cases, 2.5 million deaths. But the economic impact is huge. This is an article from The Economist, which estimates it's $10 trillion worth. And I had to look up what a trillion is, so it's 10 to the 12, so it's a massive economic loss, and as you can see, they project, the World Bank predicts that it's not a U-shaped recovery that will catch up with what we have lost, but it will be a permanent loss, certainly going forward. If you look at the impact on poverty, you can see over the last many years, global poverty reduction has been consistent, reducing, and you can see over the last year, increased by 88 million. So the impact of this pandemic has been colossal in many, many respects. Now, we also cannot say that we didn't see it coming. I mean, there have been many examples of major outbreaks, zoonotic outbreaks coming from animals to humans over the last number of years. We dodged the bullet with the H1N1 pandemic because it spread, but it was mild. With SARS, it was severe, but it could be contained, but sooner or later, we were going to get hit, and that is what happened this time with the coronavirus. Now, of course, there have been efforts made to sound the alarm, and particularly after the Ebola outbreak, there were a number of reports and commissions, including this commission from the National Academy of Medicine in the United States, in which Dean Gabriel Leong was one of the members. And you can see some of the recommendations from that commission, where they estimated, not just Ebola, but they estimated the annualized economic loss from epidemics to be of the order of about 60 million annualized basis. And they suggested, therefore, that at least investment of 4.5 billion should be annually spent in strengthening the defenses worldwide, which means upgrading public health systems of low and middle income countries, enhancing the WHO's capacity to respond, and importantly, investment in research and development. They also pointed out, and again, we know this, the failure of the current pharmaceutical industry model in developing countermeasures for emerging infectious diseases, because clearly, which commercial company is going to invest in countermeasures, vaccines or drugs, for viruses that may never be a problem? So they recommended central funding for this, and indeed, that did come to pass. So with the initiative of the Norwegian government and other funders, this organization called CEPI was set up, which is a coalition for epidemic preparedness innovations, which that was around 2017, and they started identifying about 8 high-risk threat pathogens, which included a coronavirus, 2 coronaviruses indeed, MERS and SARS 2003, and of course Ebola and Nipah and other things. And indeed, that initiative started development of countermeasures for MERS, which really helped when SARS coronavirus 2 hit. Now, I could also, when we talk about these numbers of billions of dollars, and we talk about 4.5 billion, it is interesting to compare it. For example, the cost of an aircraft carrier is 13 billion US dollars. This is just the cost to buy it, if you wanted to buy it, not to run it, okay? So we really have to put in proportion the costs, estimated costs of prevention, compared to the costs of that nation spend on other things. And to me, it boggles the mind. Right, so what can we do about these emerging infectious disease epidemics? What are the options for response? One, of course, is early detection in the humans, and I think this relates something to what Sophie was asking about earlier. So have the capacity for early detection when there is a spillover to humans, so that we can try to contain the outbreak or at least delay it or mitigate the spread. And quite importantly here, this is not just a matter of technology. It's not just a matter of smart technology and sequencing and all this stuff, which we do have. The biggest problem is recognizing that you do have a problem in the first place. Recognizing that there's an unusual outbreak and that needs to be investigated. So this is really alertness at the clinical and epidemiological level. Then, of course, non-pharmaceutical interventions. So particularly for respiratory pathogens, as was pointed out in the previous two talks, we really know very little in terms of how they are spread. I mean, the contributions before have enhanced our knowledge to this, but at the end of the day, we still know very little about how these the respiratory viruses are spread, and therefore we don't have good evidence to say that one intervention works or does not work. And indeed, that is why 5 years ago, our theme-based research program was exactly focused on this topic, and you heard some of the efforts from that program just presented just now. But when this pandemic hit, that really was all we had for the first whole year to try to contain the pandemic. Then, of course, we have what are called platform technologies, which are particularly for vaccine development and for antiviral development, which are platforms which can be switched from one virus to another. And indeed, that was the success of the current COVID vaccines. So the RNA vaccine platform was developed already for influenza, for MERS and other things, which was rapidly switched to COVID. The adenovirus vectored the vaccines were also developed for other things and was switched over. So in terms of vaccines, really it has been a great success story. But in terms of antiviral drugs, I would point out that up to now, we really have one maybe and two maybe. And those are not antivirals, they are really immunomodulators, that is dexamethasone and IL-6 inhibitors. So really the antiviral area has been quite disappointing and slow. Then, of course, we have other options to reduce the chance of these crossing over to humans, and I think this was discussed a bit in the earlier talks, which is controlling at source, and I will illustrate this with some examples in a minute. And of course, trying to anticipate what is coming so that we can preemptively develop antivirals and vaccines. And this would depend on surveillance, and again, we discussed this, that is scanning the interface at the animal-human interface, the genomics, the phenotyping, and the risk assessment. So in terms of these types of broad platform technologies to reduce, to treat patients, for example, this is work done by Michael Chan in our group, trying to understand how viruses cause severe lung injury. So what he was able to show with the H5N1 influenza is that that particular virus, once it infects the alveolar spaces, leads to a blockade of some of these sodium chloride transporters, which pump out fluid from the alveolar spaces. And when these transporters are blocked or poisoned, you have accumulation of fluid in the alveoli, and that's what leads to the lung damage and ultimately to death. And he was also able to develop one intervention, which is mesenchymal stromal cell therapy, which can reverse this. And at the moment, he is using this in vitro model, which he has developed, to try to look at other small molecule interventions to reverse this as well. Then examples of controlling these zoonotic spillovers at source. We could look at the example of avian influenza and what Hong Kong has put into place, understanding that live poultry markets are an amplifier of these viruses, and for example, intervening by rest days and by stopping poultry being kept overnight, you can see that the virus carriage in these markets essentially has been almost disappeared. And as you might or might not know, although Hong Kong was the place where the first zoonotic outbreak of avian flu was detected in 1997, since then, we have not had a single locally transmitted avian flu severe case in Hong Kong. Another example of this, and this is work from Huiling Yen, looking at these live poultry markets, she was doing air sampling at different places in the market and identified that this apparatus, this defeathering machine, is one of the major sources of aerosol generation. So when the chicken is killed, it is put into this, which spins at high speed and removes the feathers, but obviously it's a great aerosol generating machine. And this, collaborating with people in engineering, they showed that the aerosol generation does come from this procedure, and they then developed a fairly simple modification to this, which reduces the spread of aerosols from these defeathering machines. And then, of course, finally, work by Guan Yi and others and his team, where I think Vijay was also very involved in this, essentially showing how these zoonotic viruses emerge, whether it be H5N1, H5N6, H7N9, they follow a common pathway with viral gene segments coming from the wild bird reservoir through the domestic duck pool, and then reassorting with viruses from chicken, mainly the H9N2 viruses. So by, in theory, separating the marketing of aquatic birds with terrestrial poultry, you can reduce the emergence of these viruses. So these are examples of, without trying to predict which virus may be the next pandemic, at least in the case of influenza in this example, you can, by understanding the pathways, you can reduce risk. And then finally, how do we know what may be coming, or how can we anticipate what is coming? It was just 2 years ago, I think, we had a Keystone Symposium here, and they recorded a session which included the Marianne Koopmans, in fact, and myself. And one of the questions that was put to us was, what is the next pandemic, essentially? And to cut a long story short, the answers were, at that time, influenza and coronaviruses. And thirdly, of course, paramyxoviruses, Nipah as an example, but that whole paramyxovirus group. And of course, that turned out to be true. But then how do we exactly identify, as the very first talk, Zheng Li was talking about, say, within the coronavirus group, how do we identify which of these viruses we need to pay attention to? And that, of course, is probably even more important than just finding the viruses. And here we have the example from influenza, where again we have a situation of many, many viruses out there in the animal reservoir, and how we risk assess it. I don't have time to go into that now, but this is now being used by WHO. And you can see that sequencing is there, but it is only one of many parameters that are looked at, including spillover to humans, which is what Sophie mentioned, transmission in animal models, and, for example, looking at tropism, infection of these viruses in human ex vivo lung, again, work from Michael Chen and others. So with that, I've just come back to the same slide. So these are the options we have for a response, and I think we really need to invest much more than we have done before the next epidemic or pandemic hits us. And I just also want to end by saying that pandemics are bad, but really we need to use this pandemic as a learning opportunity for tackling some of the other global threats, which are issues of equity, as we see from the current vaccine problems, the degradation of ecosystems, and of course, climate change and environmental pollution, which are much more insidious, but in the long run, will probably be much more damaging. Thank you.
Thank you for the excellent presentation, Malik. I'd like to open the floor, as well as the panel, if there's any points that you'd like to raise. There was a hand behind you.
Yeah, I'd just like to ask the panel a couple of questions. Also, perhaps Dr. Sher, if she's still online. We see that in some of the viruses presented, for example, one of the most common being the RATG13. It's been in a lot of papers, which is touted as being kind of 96% identical to the coronavirus. But speaking of DNA, we know that even a small variation of 1% can be a chimpanzee or a human. So when we speak about 4%, this is actually a very large number. So how much confidence can we be sure that the virus that we're dealing with as novel COVID-2 actually comes from these bat coronaviruses? Just as a last point, our present speaker just noted about the H1 viruses, H1N9 and so forth, are coming from wild birds to ducks, then mixing with viruses and then ending up in the chicken population. So I also wondered, as a virologist, or those who are here, is there anybody doing some studies to try to mix a few different types of viruses together? For example, the current the RATG13 with other coronaviruses to see if they can make a higher match in terms of matching something that we see as kind of nCoV-2.
Dr. Zhengli, I was just wondering if you heard that questions.
I'm sorry, can you repeat again, please, your question?
Yes, of course. Perhaps if it's a little bit too noisy, then maybe the moderator could assist. The question was kind of two-part. First being, 4% difference in DNA is actually quite large when we compare other types of differences in DNA material. So therefore suggesting, how certain can we be that the virus comes from the bats, the horseshoe bats or others that we find that are hosting the coronavirus? And then secondly, is there any viral research being done? Yeah.
You can wait till the first question is answered.
Sure.
Dr. Zhengli, the question is, the virus for example, the genomic difference between RATG13 and SARS-CoV-2 is about 4%, which is huge compared to the scale of evolution. So the question is, how certain can we be that these viruses came from those hosts that we now understand it most likely came from bats, for example?
Based on the current data, we are not sure if the SARS-CoV-2, if the T213 is the most closest ancestor of SARS-CoV-2, because even though the T213 is the most closest based on genome related similarity, but in fact, based on the receptor analysis, the T213 is not T213 spike protein has a very low binding affinity to human ACE2, much lower than the coronavirus found in pangolin. So based on the receptor usage or the binding affinity data, we are really not sure if the T213 is the most closest ancestor of the SARS-CoV-2.
You want to go ahead with your next question?
Thank you for that. So kind of similarly, then the question would be is, how can we answer for this 4%, 3% difference that we find? Could this difference come from the merging of two different viruses, something that we've seen in our recent talk between the kind of H1N1 and other viruses that we're receiving from the poultry population, whereby we're mixing one coronavirus with another to produce the novel coronavirus that we see today in the pandemic?
I mean, the next question, I think, is a bit of a tricky question. The question is actually whether the SARS-CoV-2 originated by recombination of two different coronaviruses, and is that a possibility?
Yeah, based on the previous knowledge, so coronavirus, tends to be recombined if the two different coronavirus infect in the same cells. And another evidence is based on bioinformatics analysis. But I think the recombination exists for the coronavirus. But based on the current sequencing of the SARS-2 related coronavirus, I'm not sure if the SARS-2 is a recombination of different coronavirus, because we need to find more sequencing to fill the gap and to understand better how the SARS-2 came from. So I think I'm not uncertain about this question.
Okay, that's wonderful. And sorry, just do you have any things that you could speculate on based on some of the research in the field about what recombination might be occurring, what other viruses might be responsible for this?
Can I just, I mean, if you just take the story of SARS-1, okay, so SARS-1 outbreak happened in 2003. The bat ancestor was detected in 2005 by Zhang Li and from KYUN's group. But then it took another 8 years to really find the close ancestor of SARS-1. And that again comes from Zhang Li's work, right? Because there's a huge diversity of coronaviruses out there in the bats. It would be quite, I think, unreasonable to expect that we had a virus emerging last year that we would have an exact match in bats by now. I think it is quite possible that as people like Zhang Li look at the diversity of bats out there, they will find a much closer match to the current virus, just like was found for SARS-1. Of course, it could be recombination or whatever it is, but it is, I would suspect, very likely that it is out there in nature. We have to look. I mean, there are millions of bats and thousands of species of bats out there.
I mean, I'd like to just add to that point. I mean, so we've been doing influenza surveillance extensively in multiple hosts over several decades now. We had a new virus in 2009. We were able to roughly tell what it was. But to pinpoint where the origins were and what viruses mixed to form that virus took about 6 to 7 years additional surveillance. So these things are not very easy to do. And I think that's the conclusion.
Actually, I have one question about this. So I think your questions is based on the assumption that if you see an emergence in the human, you expect a higher chance of detection of a very similar viruses in the bat populations. But I wonder that maybe you two can answer, or Professor, whether actually a virus very adapted to be in the bat population, very highly transmissible in the bat population, will be as more transmissible in the human population. So what I mean is that, for example, maybe there will be a lower chance of finding this viruses very close to the human in the bat, just because this virus is more adapted in the humans but not in the bat. So I'm just not sure whether the assumption is valid in the way that you expect it's quite easy to find a very similar strain in the bat, just because it emerged.
Based on the work that Meredith has presented, it's possible, or it suggests that maybe the emergence of our pandemic today is actually the recombination or the merging of two different viruses. And I just wanted to know, what is this likelihood? And that, seeing that we might actually not find something. Of course, I respect this. I mean, I'm doing research myself, as we all are. We know the time it takes to conduct our bench work and genomic sequencing and so forth. But I'm not a virologist, so I thought it seems a possibility that I haven't heard people discuss, that it might have been 2 different viruses or others. Obviously, Dr. Sher just noted that it's likely not 2 coronaviruses based on what we know today. But yeah, I thought it might be something of this nature.
Yep. Unfortunately, the viruses evolve too quickly, even in their natural reservoirs, that we can't maybe never be able to trace back the exact origins of this particular virus. So now I'd like to actually turn the discussion points very quickly towards Malik's talk, and if there was any discussion points. And first, I'd like to check with Tamara if there was any online questions as well.
No questions online, but actually I had a question for everybody, just to sort of rebound on some of the thoughts that Professor Perez had. I was wondering where you see sort of lay publics in all of this. For instance, Professor Perez, you suggested, that yes, you can come up with measures that would be effective in sort of preventing, widespread transmission, but in fact, you need public buy-in to actually accept those, or to go along with these measures. And also thinking about public practices for helping to identify the most likely candidates, for the next spillover, for the next pandemic, beyond, sort of large categories of, okay, there's deforestation or there's urbanization, but looking at very specific, fine-grained, systematic studies of the kinds of practices that, for instance, duck farmers or people raising bats or others, how they're thinking about these animals and what their specific interactions and thus risk factors are. So I'm wondering where you stand on that, whether you think that that's important or whether that could contribute to your investigations, and if that kind of work has already been done.
Yes, so in regard to public buy-in, I think there are many different levels at which we need public buy-in. First and foremost, the public and political buy-in to invest in some of the measures I talked about, such as countermeasures, vaccines and antivirals in anticipation of possible pandemic viruses. So at least if you have potential vaccines for a virus family, we might be able to extrapolate that approach would work when another virus from that same family causes a pandemic. Indeed, I mean, this time, COVID-19, the vaccine development was helped by investigations that were done after the SARS-1 outbreak. I mean, the fact after the SARS outbreak, there were attempts at vaccine development. It was clearly shown that the spike protein is the key target for protection, et cetera, et cetera. So all that work did help in response to SARS-2. So similarly, for example, in the case of the paramyxovirus family, which is again another big group of viruses, which things like Nipah may be potential threats, I think we need that investment to anticipate things that might come. And also similarly, antiviral drug development, which can be quite broad across a particular family. So that is on one side. Then, as you pointed out, the other aspect is behavior modification to risk reduce the chances of these spillovers taking place, and I completely agree with you on that. So for example, you can have the example of, in the case of influenza, the risk of live poultry markets. I mean, I gave you examples of how the risk can be reduced, but maybe ideally, we should get rid of these, although I realize that this is has got large traditional cultural involvements. Similarly, for other zoonotic jumps, I mean, certainly in the case of SARS-1, we clearly know the interface that the virus jumped from animals to humans was these game animal markets, the wild game animal markets. And indeed, after that, the closure of those wild game animal markets in Guangdong prevented the reemergence of SARS, because the virus was hanging around in those animals. Now, again, whether we can enforce the closure or drastic reduction of this wild game animal trade, which is present in a huge part of Asia. I mean, and it's not only a risk for pandemic emergence, it is a major threat to biodiversity maintenance, because we have many animals going extinct, for example. I mean, pangolins being one of them. So I think identifying some of the critical points at which we can intervene generically, and then, of course, how we get the public to buy into that is, I appreciate, is a big challenge, particularly because these risks are very stochastic. So if you take avian flu, H5N1, for example, in many parts of, during 2004, 2005, many parts of Asia, the virus was found almost in every live poultry market. But hardly any poultry worker got diseased. So how can you, so people can't understand how this can be a risk, because I don't get sick, right? So these are some of the challenges we have in trying to implement some of these interventions, these risk reduction interventions. Over, but other people may have some other thoughts.
Maybe we can quickly hear a couple of thoughts as we exceeded on time. Is there any questions?
There's one other question here. Can I read it out?
Oh, please go ahead.
Okay. This ties into the question of public buy-in, but misinformation online played a larger role in hampering public health measures than most people foresaw. How can we prepare to combat misinformation for the next pandemic? This was from someone online.
Right. I'm not sure I'm the best person to answer that. I mean, maybe KG can comment on it. But, it's wrong to say that it was not foreseen. I mean, if you watch that movie Contagion, which I think was an excellent movie about 8 years ago, made on the next pandemic, this disinformation was highlighted in that movie absolutely. And I think this is a problem that today's society has to contend with, not just for pandemics, mind you, for many, many things. But KG, can you give us some points of wisdom as to how we tackle this issue?
No, thanks. I was actually asked by some students, some journalist students, the same question yesterday. And I pointed out, if we're talking about outbreaks, the kind that we've been dealing with for decades, it's always so confusing at the beginning because we have minimal facts and we have maximum speculation. And then we have a lot of spokespeople. And that is a combination for confusion from the very beginning. And that remains true now. But I think your last point is really the key point, is that we have entered into an era where the mechanisms and the use of misinformation have not just arisen because of confusion, it's used purposefully. And we have social media and we have platforms which amplify that and allow it to expand. It's like having a large electronic amplifier. I think that this was worried about, and it's always worried about at every outbreak that we've dealt with. I think the communications is generally the most difficult issue we have to deal with. And to be a little bit blunt, we don't have an answer for it. We simply don't have a good answer for how to deal with this level of confusion. And partly because the previously accepted sources of authority like WHO and various organizations themselves are not regarded in the same way by many people. And this is a very, very big issue, and I'm not quite sure how to deal with it.
On that note, I'd like to conclude the day one proceedings. We've had excellent speakers and excellent audience, actually. I'd like to thank the audience first, because we've exceeded nearly 1 hour and nearly 100 people have stayed online and here watching the talks. I'd like to thank each and every one of the moderators, the panelists, the keynote speakers for making this occasion a fantastic event and raising various interesting discussions and topics on origins and transmission and control. Please come back tomorrow because we've got Marco Vignuzzi from HKU-Pasteur providing us a keynote lecture, followed by a panel discussion on disease surveillance and enhancing virus surveillance. And we have speakers from across Asia, Africa, and Europe. So please be back tomorrow. And thank you again for attending this today. Thank you.